So we're going to go ahead and get started. We had over 40 people sign up, so I'm very appreciative of those that are in the room and chose to come here instead of seeing this beautiful sights in Halifax. Um, so we might have some more people enter the room as we go along, and that's fine. With the small crowd, we can certainly do. JJ said we can do one-on-one teaching, but feel free to ask us questions. I still think hopefully we want to make sure this is useful for everyone that is here. So just in case, make sure you're in the right place. Can everyone hear me okay? This is the course on benefit risk, and we have several guest speakers who are experts in the field that will cover different topics. My name is Alicia Gilsonen. I'm Vice President, RTI Health Solutions. I've been the course facilitator for this for several years, the past co-chair for BRACE SIG, and have had the pleasure of working in this field for several years, mostly doing some training on the BRAT framework and qualitative framework. But today I'm going to leave it to my colleagues to give the meat of the presentation, and I'll let them briefly introduce themselves and where they are from, just to give you some context of their talks. Derek?
Okay. Must be more important than me. Too much is working. I'm Tariq Hamad. I'm currently the Vice President and Head of Medical Safety of Market Products in Takeda. And I used to be in the FDA for maybe 13 times. I spent most of my career as a regulator. I have been involved in benefit risk since the beginning. I was part of the ICH guidance group that was negotiating the revised guidance. So hopefully I can give you some insight on what happened building this guidance.
Hi, I'm Bennett Levitan from Janssen or J&J. I lead a team that does benefit risk, patient preference studies. We also have been involved in a lot of policy and methodology work over the years, and you'll see some insights, not just on the technical side, but how the work connects to the FDA and EMA as we talk.
Thanks, Bennett. I'm Johari, but call me JJ. That's how I'm called. I head epidemiology and benefit risk at Sanofi. At Sanofi, we do benefit risk evaluation in a very systematic way. It's anytime we go to decision making, we must have benefit risk evaluation, so we could provide that practical perspective. I've been involved in different public-private partnership as well. The last one, we just finished actually the draft of CIOM's 12 working group on benefit risk evaluation. So you may have seen that it's been reviewed. But anyway, look forward to discussing with you guys.
And we had a fourth speaker, Dr. Carol Mansfield, also from RTI, who is head of our patient preference group, that was going to speak today, but her flight was canceled and she was unable to make it. So Bennett has graciously agreed to give the presentation on her behalf, and is we appreciate that very much. So before we get started with the rest of the program, I walked around and tried to get a sense of the attendees in the room. So it sounds, how many of you have had some awareness of benefit risk assessment in the past? So some people have some knowledge. How many have done a benefit risk evaluation or contributed to a benefit risk evaluation? We have 4. Okay, how many are from industry? And do we have anyone from government agency? Any students? I know that people might raise their hand twice. Did I miss anybody? Their perspective? Academia, thank you. And how many people know what patient preference studies are? I've heard of patient preference. A couple. Has anyone been involved in designing or reviewing results from a patient preference study? A couple. Okay. So today we will be talking more about Tara will give a background on kind of why benefit risk assessment is important and start introducing the patient engagement is becoming more and more important. So there's the benefit risk assessment process, and we like to think some of us that the patient preference is can be a component of that, but it is different. So as we transition from talking about the overview of benefit risk assessment into the patient preferences, just keep that in mind and feel free to raise hands if you have questions along the way. And JJ will give an overview of the structured benefit risk assessment and some considerations. And I think you're going to touch on qualitative as well, just a little bit. And then Bennett will give Carol's talk on patient preferences and then also wrap up trying to put everything together. This is our tentative planned agenda. If we stick to this agenda, you'll see that we'll be ending a few minutes early, and that's really up to you guys. Not trying to get us out early, and our speakers, but we would like for you to stop and ask questions along the way. If you do, we don't want you to be lost. If you can't hear us, can't understand if we're talking too fast, please let us know, because I know it's very frustrating to be sitting in the audience and not be able to follow along. The coffee break we have tentatively scheduled for 3:45. It is confirmed that it will be there. So if we are running into a little bit late, we'll still give a coffee break. And in case you didn't notice, it's downstairs by the registration desk. And at the end, we have some questions that we'll be discussing with the panel members that are really relevant to how does pharmacoepi fit into this whole framework of benefit risk and patient preference. And we'll have more time for question and answer at that point. So this is just a reminder of the coffee break. And I think, so we can't see the slides while we're sitting at the table. So I'm going to ask the speakers who are not presenting next to sit in front, and then we'll rotate through here.
Hello, everyone. Thank you very much for coming. You should have listened to Alicia's advice, because I actually surprised. I have few quizzes I'm going to ask by people at the back. So if you don't want to be quizzed, you should come closer. So anyway, so today we will talk about of course, after the disclaimer, this is just my perspective. It's not my employer. The objective really is to help you appreciate why the whole benefit risk structured approach actually was created or was even discussed, and it all started by the ICH, and actually help you appreciate the intent behind what was the discussion that happened there, because I was a member of the negotiations. So not everything ended up clearly enough maybe in the guidance. So this can help you appreciate what is actually intended in the use, and how the regulators are actually using it now to help you appreciate also the value of the structured approach. At the end of the day, the idea is that, of course, you don't have to listen to anything that I'm saying or my colleagues, but there might be some lost opportunities, because the new guidance is opening the floor for other options that you can be utilized to tell the story about your drug, kind of thing. Now, how many of you are familiar with this movie? This is a very old movie, A League of Their Own. And the idea is that this was a women's baseball team. They were still new, and they were not sure, and they were supposed to go on a trip, and they were standing in a station, not really going on the train. So he said, well, one of the actors said, the trainer, are you coming? See how it works? The train moves, not the station. So the point is, the train of benefit risk not only will move, it's already moving. And if you are not on the train, you will be left behind, because as you will see, the regulators expect it, everybody is expecting it. So let's start by some of the regulatory landscape. Now, can you see this picture? Do you see what's happening here? So this guy is trying to fix the AC. He's upside down. Now, do you think this is crazy? Looks crazy to me, you know. So anyway, so what are the benefits? Maybe better temperature, The risk is, of course, fall, serious injury, or death. Now, what I'm going to force you to take sides. Who actually thinks it's crazy? Just by show of hand. Okay, I have 1, 2. Oh, right, great. How many of you think it's okay? Or the jury is still there? Oh, nobody. Okay, no, somebody was scratching, but it counts as a vote. No, but you're the only one right, actually, even unintentionally. Why is that? Because you have no idea about the other factors that is helping them decide what is the benefit risk. Maybe the building is not that high. Maybe there is a safety net. Maybe there's weather is really very bad and it's worth the risk. And I remember actually the first year I was in the FDA, one of the divisions approved a drug with osteosarcoma signal in animals. And almost everybody in the agency was up in arms, like, how can this be? Why would you do that? And they actually ended up having to do a whole seminar to inform the agency, the rest of the teams. Oh, we did it because of this. We had risk mitigation, and there is no other drugs. And so they had to like explain themselves basically what were they thinking approving this drug. And that's exactly the idea of the structured benefit risk. It helps you tell the story about what are you thinking, claiming that your the benefit of your drug is overcomes the risk itself. So, but some of the resistance that you will find in your companies and among your colleagues saying, well, I mean, I've been doing this for like 40 years, and I quote, this is the first thing I heard, like maybe 10 years ago when I was starting to think about this and do it. So what's new? There's nothing new. The issue is that there has been, of course, the benefit risk is has always been and will always be an integral part of the development. But the idea is that the regulators were complaining, oh, it's not really taken seriously, the section of benefit risk in the submission. And in the very first day of the ICH meeting, the FDA brought 14 benefit risk sections. Some of them were 2 pages, some were 20 pages. And they were telling us, see, I mean, nobody's taking it seriously. Of course, I told them that's the reason is because they have no idea what you guys are doing with this section. So it's confounded judgment, and they agreed. So the other also people were complaining, the industry was complaining because they say, well, what is wrong with you regulators that we give the same data to EMA and they are fine with the FDA, they have no idea, they don't agree, and we have no idea what is the basis of that. So it's just a matter of knowing the structure and the thought process, understanding the reasoning behind what's going on. So we ended up meeting several times. The last one was in Lisbon, where we finalized the ICH guidance. And this actually started making the discussion actually to be taken more seriously about the structured approach, not the benefit risk as a concept. And we ended up with a simple structure, but it captures all the needed elements. Now, we spent 2 years doing this. You might say, wait a minute, you spent 2 years and that's all you got? That's too little. Well, the reality is this, of course, behind this was like 5 pages of explanation and factors to consider. But also, this is a consensus meeting, which is everybody has to agree on every single word. It was difficult to really add too many things. And that's why actually I want to give you some of the insights about what's intended behind various sections. And also some of the other guidance, the more recent ones, FDA, they went way beyond what we thinking they will go. In 2017, the EMA adopted it, and even in the CHIMP work plan for 3 years since 2018. Actually, I did not check the one for this year, but they always reiterated some of the concepts that were introduced in the ICH guidance, which is the methodology of benefit risk and the quantitative piece as an option for you to consider, which is something that JJ will talk about some more about the methodology, and also the patient involvement, which is what Bennett will be talking about. So this has been actually being repeated again by the regulators. So, but I have not seen that many actually. I work in several companies now. Not everybody is taking advantage of this option. That's where the lost opportunity is that I started with. So what about the FDA? The FDA actually started even before the ICH finished. We had an Institute of Medicine workshop where they examined the sources of uncertainty in benefit and risk. And I have here the actual link for you. I had a talk in it discussing the causes, but the link covers the whole workshop if you are interested in understanding, because the uncertainties are expected to be part of the structured benefit risk now. And then like 6 months later after EMA, the FDA also took on or adopted it, and they were actually the chairs of the whole group. Now, around that time, the FDA started a pilot where they actually providing the feedback to companies in a structured way. And the idea here is that, as I said, your job is to help the reviewer help you. That's another way of losing opportunity. It's not just about not doing something you could have done, but also about presenting your data and packaging it in a way that will make it harder for the reviewer to find the information. And as a previous reviewer myself, this was one of my pet peeves, really. It is this, we are way overworked, and to hunt for the piece of data was always one of my pet peeves. Think about it. I mean, I had managers, and I had to be able to defend my decision, either yes or no, or drug is good or not, kind of thing. I'm sure you have seen this structure before. So what I've seen in some companies do, they actually provide some of the summary in this table structure, because the regulatory reviewer actually sometimes provide it back in the same structure, so to make it easier to find the information. And in 2021, they actually, FDA published a more detailed guidance. Of course, it's a draft. Some guidances stay draft like forever, but I'm not sure if this one will actually be finalized. I would imagine it should be finalized by this year. And they actually are also referencing the 2016 guidance. And I will touch on some aspects of it when we go in the actual parts of the guidance itself. And even in the PDUFA 7 commitment, again, the FDA is still reiterating the patient voice and the methodology and the way things are being structured. So the commitment is clearly there. That's why I'm saying the train is already moving. So if you are not, or your institution is not on board, it might be even too late now. So you have to be on board quickly. The last piece of the puzzle for the guidance is the SIMS 12. It was out for public consultation in May, June. I'm not sure when it will actually be finalized. Maybe JJ can tell us, but soon it will be finalized. It takes a while to adapt. I was part of SIMS 10 and took us like several months to accommodate all the comments. So it might take a few months or something to finish. But you will have so much guidance out there that it should help you. So now, let me give some insights on the actual sections of the guidance. If you think about it, there are some logical pieces of the process. You have identifying the key benefits and risk, and then you do the actual assessment of the benefit risk. And you do this within a context, which is the therapeutic context itself, the disease itself, and the current therapies that is competing with your drug. And if they added some sponsor activities actually to that guidance, so they expected the kind of the general structure, but they added few more aspects as you'll see. So let's start by the therapeutic context. That's the first thing you start in when you are thinking about your drug and how you will be telling the story of your drug. You have two pieces here, two parts: the disease itself, or the current therapies. Now it's time to check your knowledge. That's the first quiz. Whether you have to figure out what's fake news and real news. I'm not sure if you follow the US elections, but this actually is coming back in light with the return of the previous president. So now let me know what you think about which one is right or wrong answer. Now, which of the following sentence is incorrect, basically fake news about what should be included in the therapeutic context section? What do you think? Which one of those, A, B, or C, should not be in the therapeutic context? Now, you might, some of you, most of you said you have some experience, but even if you don't, just logically, which part should, I will give you like a few seconds, and then I will ask you to vote to commit to one, A, B, or C. And nobody scratches, otherwise it will count. Okay, so how many of you vote for A, that you should not include description of safety efficacy of your drug and the benefit risk section in the therapeutic context. Okay, I can see one. I have one vote. How many of you says it's B? Huh? And even the coffee sipping counts as vote. And then see how many of you vote for C. Okay, so you have a couple of it. So that's good. So it's a equal distribution if you would. However, the correct answer is what Victor mentioned, checked, which is the efficacy and safety of your drug. Remember, this is a therapeutic context. And basically, you are preparing the context for what is coming, the results of your drug. Now, the value of knowing this and having it clear in your mind is that you can actually start this much, much earlier. And as pharmacoepidemiologist, you will have significant role, as you will see, in building up the case for your drug way ahead of time in parallel while the trials are taking place. So basically, this section is agnostic to the new drug. That's where you can think about subpopulation that might be relevant, and then the limitations, uncertainties in the field. That's where your role can be very significant. Actually, you can actually help the clinical team appreciate how can they position the drug in the discussion eventually. So the idea is that, of course, you touch on the greatest relevance and impact to the patients of interest, and then you don't have to cover the whole indication of the whole disease entity as much as just the narrow indication, because it might be tempting that to cover everything around the particular indication, but you have to focus only on the indication that you are trying to make the case for. And then the societal and public health implication. All these parts of what we propose to take place in the guidance, as I said, that's where epidemiology and real-world evidence can play a role. However, think about it, this will take time. And if the team is not prepared and planning ahead, it might take, people might not really, it might be a lost opportunity, as I said, because it will take time to search the literature and figure out what is the impact and how to frame it. Now, another quiz for you. In the current therapy section, now what's fake news? If you can read it quickly and tell me which one. You have limited information to unmet needs, describe or relevant needs, met or unmet, simplify providing commentary on the class, and then measure difference between regions that may be noticed. So which one is incorrect? So how many of you, I'll give you like a few seconds. How many of you will vote for A, that this is the incorrect one? Nobody? Okay, I have 2, 3, okay. How many vote for B? Huh? Nobody? So how many vote for C? Now what about D? Okay, so I have some votes. Okay, so it's more consistent this time. So the fake one is the A, to limit it to unmet needs. Why? We had a lot of discussion about it in the meeting, and as you will see that there are some Me Too drugs, so it's okay to discuss the medical need. Doesn't have to be just unmet. So we were careful in phrasing it to make sure that people don't get put off and they have obstacles in when they are trying to figure out how to make the case for the drug. And the idea here is that you can include even off-label interventions if you can find in the literature and supported or by expert opinion. You can include non-drug interventions like diet modifications or physical therapy, whatever that works applies to your situation. You can, if you have some classes are huge, so you might summarize by the drug class, what's known about the class itself. And if there are any regional differences, this is your chance in this section to highlight those. So that's very important to put in mind. And you will have all these slides as handout, so it will help you to go back to if you are interested. So the idea is not to compare and contrast between, it might be tempted to say, well, you see, here is the uncertainty, but my drug is better, and so on and so forth. This is not a place to compare and contrast between your drug and whatever else is going on. So now the next quiz, In painting the benefit risk profile, all benefits and risks should be incorporated for transparency and, of course, to be aligned. You cannot hide anything, right? Is this fake news or real news? How many of you says it's fake news? How many of you say it's real news? Nobody's committing. Oh, okay. As nobody is scratching. I can see you, and the coffee is gone. So, well, it is actually fake news because not all drugs, not all. You have to focus on the key benefits and risks. Not all drugs, not all. I'm sorry, benefits and risks are equal. Can you figure out what's the difference between these two? They look different. I'm sorry, they look the same. I mean, however, how about now? So you might find some risks or benefits not really fitting to be as a key risk or key benefit. So the idea here is that this needs a critical evaluation. That's another role for pharmacoepidemiologists to try to figure out how can you defend and make the case for something is key or not key based on maybe what's in the background, the public health impact, and whatnot. And the guidance is providing many factors to consider for the evaluation. But I mean, again, because I work in several companies, it's not really done consistently. Some say, "Oh, we're not gonna just bother with it." Most of the time, because it's thought of too late, and again, it needs effort upfront to be able to make the case for why some risks might be more important than others. So now the next quiz. Only primary study endpoints as well as information eligible to be in the drug label can be used to frame the benefit risk profile. Now, how many of you think this is real news? Huh? How many think it's fake news? Oh, so again, man, you must have attended this talk before, unless you have seen my slides, because it isn't. I uploaded all the slides. So this is actually fake news also. Why is that? Because okay, I will tell you later why is that. So it's not just the primary endpoint. Now the second point here I'll try to make by another simple quiz is what do you do when you describe your benefits and risks? Now there's no quiz there. You have to verbalize it now. Can anybody tell me what you see here? Let's describe it. Let me see. Okay, that's one description. Anybody more optimistic? Aha, I got you to say the wrong answer. That's good. Yeah, that's successful. That's success. The point is, I did not ask you about your opinion and judgment of it, although I alluded just to make sure somebody get the wrong answer. The point here of the guidance is to be factual as much as possible in all these sections before you get into making judgment. So when you are talking about the benefits in the description, you are being factual, providing factual summary, and you are talking about not just primary but also other endpoints, not just that additional one. And then when you are talking about, you can have a surrogate and talk about the surrogate. However, you have to be careful because in using a surrogate, you have actually to try to make the case for how much it predicts the actual clinical findings and make sure that this is the case. Also, you can even mention convenience if the drug, your drug is providing more convenience, if the dosing is less, for example, if there is any issue around compliance with other drugs. So you can utilize anything that do you have a question? Yes, yes. But see, remember, many of the sections I mentioned, you can prepare early. So that's how you are preparing the thought process. And at the time, this might actually help you appreciate that maybe I need the patient preference study. Maybe I need to consider quantitative assessment early on in the process. It's not about identifying those early. Remember, this is for the actual submission. So when you get there, when you actually know, then you'll be able to make the case and be convincing. But the planning has to start much earlier, and you are right. and you get ideas about what you might need to use the time. Because if you wait until the end to say, oh, but I can't do this, you can't do that, nobody will gonna wait on you for like 2-3 months and lose the opportunity for submission. So that's another lost opportunity kind of issue. So now for the risk, the same thing, being factual, but also the idea of the guidance says associated with the risk has to be associated with the product. Now the question is, what do you do with the potential ones? Are they associated or not? I mean, technically, you don't know yet. You are not sure. You suspect. We had a lot of discussion around that, but we ended up having this as an option. It's up to you to make the case for whether you want to even include potential risk as key or not. So it can be key in some scenarios. And also, so the point I was making, well, what about what you do in emerging potential risk? Do you also consider it as key or not? So this is going to be left for the discussion based on the context of the indication and of the submission itself. Also, not all the risks are included. So if in effect, this almost creates 4 versions of your risk, because you have a benefit risk section focusing on the key risks, and then you have the economic modeling. By the way, I asked some of my colleagues, I'm not sure if this was a while ago, they use only the identified risk, don't use the potential unidentified. And RMP now uses only the important ones that you can do something about with the new changes. And then, of course, you have the actual submission and the paper and IB that you have to list everything. So, and that's one of the reasons for why some companies don't really bother selecting the risk to put all the risk everywhere, which it's again a lost opportunity to make the case for you for your drug. So that's the end of this part. But now let's talk about the overview of the benefit assessment section in the guidance. In that section, all you have to do is to explain the reasoning. What were you thinking, claiming this your drug of yours is good enough to submit? And that's why I was talking about the being factual and being agnostic at the beginning, because this section is where you put all the thought process and all the comparative aspects that you want to put. Of course, you have to include uncertainties as expected. Now, some highlights from the guidance, FDA guidance. I'm not going to, of course, go into all these details. You can. I'm actually including the pertinent details to work as so the reference for you to make your life easier to identifying the pieces that matters for you when you are doing this, putting this together. But the guidance has a section on if you have a serious risk, it's already found, how can you make the case for that drug still? So you have a chance to, if you consider this, maybe the benefit is truly meaningful. Maybe it has advantage over others. Maybe you can truly mitigate it. Maybe actually the last point here is interesting because we also spoke about it in the ICH. If the drug is acting fast, then maybe you can say, okay, we will know that those subset patients are benefiting quickly, so maybe they can tolerate the risk, and that's where the patient preference also come, if we think about it. Now, also the guidance is focusing on certain considerations, is helping you to appreciate how can you make the case for your drug, and it's talking about uncertainties and patient experience. And these are the selected parts. The guidance is long, but I selected parts to help you think through and make the case for your drug. Like, for example, they went through over many aspects to consider as general consideration for why you make the case for your drug. Again, the extent of benefits and the extent of risk and so on and so forth. So I'm not going to go over all details. You have it in the slides. Now, for the uncertainties, actually, they also are offering some sources and limitations to help you out. I remember actually when I was helping some teams, I was head of benefits in a previous company, and some teams were like, okay, how can we, this is like when you are in an interview and somebody asks you, so what are your weaknesses? You get stuck, right? I mean, you're not quite sure how to answer that without shooting yourself in the foot. So the FDA is helping by providing some aspects you can utilize to comment on the potential uncertainties without shooting yourself in the foot, kind of thing. They even have a whole guidance on uncertainties, but in the devices. I found some of it helpful also in drugs, so you can have a look at this guidance also. Now, for the patient perspective, they actually, that's one of the two notable aspects for the new options available to you based on even ICH guidance. And then all the other guidances after that is has taken it on. So the idea is that you can utilize that either directly or indirectly with the caregivers. We had to like fight to have the indirect patient perspective in the guidance itself because the caregivers for some disease entities. And the idea here is that the FDA took it further to actually go into more details and even talk about measuring the impact of the disease on the patients and deciding on the unmet needs and what benefits actually meaningful and acceptability of the risk and the value and burden. So all these fall under the patient preferences kind of field of study. So that's why actually we are linking, connecting these two together, because it is again part of the lost opportunity if you don't realize it. And later on, it's, oh, well, I thought we can do this. No, it takes months to do that. So you have to be thinking about this ahead of time. And that's why you start even early in the process, in the planning process. And just as the hot off the oven, we actually, or off the press, we just published a paper last week for the IMI 2 group on how patient preferences can be used and communicated in regulatory evaluation. So this might also be helpful to you. This was published just last week. So in a nutshell, the paper actually just to give you an idea about what the paper is about, it reiterates the value of the patient preferences in drug development, exactly like the guidance, and it tells you what it helps to inform the regulator about. Basically, it's very educational for the regulators for benefit risk. They would know about all aspects. And the paper also go over my preferred project itself and how it was put together. We have many literature reviews, interviews, focus groups, and the like, and provide actual insights and recommendations, specific ones. So I will not go over it, but you have the paper and you have the slides itself. So the second notable aspect, in addition to the patient perspective, is the quantitative analysis. That's where J&J will, JJ. Exactly. I always make so, so JJ will go over this aspect, will help you appreciate more. Now, what is this quantitative assessment? And here I'm not saying how to decide to do it or not. JJ will help us with that. I'm just helping you appreciate some of the definitions, at least from my perspective. Now, if you are talking about pure qualitative assessment, like should I use A1C or glucose level to judge on diabetes progression, for example, that's a pure qualitative approach. But basically, it's a judgment, value judgment, if you would. But if you judge and use some quantitative metrics like risk difference or relative risk and whatnot, that's what's perceived as semi-quantitative. The true quantitative ones that matters from a benefit risk point of view is the more complex modeling one, where you have both judgment and actual weights put on the benefits and on the risk, numerical weights. So if you think about it, this is the more explicit, weights are more explicit here and more implicit in this, but the weights exist basically, but one of them is more by the judgment and one actually based on actual conversation and discussion and negotiations actually. There is actually a paper published on cladribine. It was a drug that it was one of my drugs in 2 companies ago. It's good, it gives you an idea about what exactly happens with that. So I can also provide this to you. And if they finally listen to what we were trying to convince them to do, which is publish their own MCDA. So that's another precedent. So the precedent is there. So it's a matter of utilizing again this opportunity if you feel your drug has a chance. Also, I mean, just early this year, there was another guidance that was published on the quantitative benefit risk by the ISPOL group. I mean, a very good group of people actually worked on this. I know many of them. And then, I mean, if you want to understand or appreciate more analytical aspects of what are the implications, where you can, how can you characterize the benefits and risks better, I published a paper just after the guidance. You also find it in the literature. So this can also be helpful. So in conclusion, and I think I will be right good on time, right? Okay, that's good. So just remember that this whole field is still evolving, because it's not just the fact that 2016 is too old, everybody knows about it. The challenge is in the application and utilization of these opportunities, which is not clear until now. The complaint actually that people have is that some regulators don't even tell you if they utilized what you provided or not from those more sophisticated approaches to benefit risk. So it's matter of really finding out the how to best maximize your the value of your effort. And of course, the consensus was reached and to standardize, and everybody is on board now. Just remember the notable aspects that there are things you can do, go beyond your primary endpoints. Remember the patient experience and the quantitative benefit risk as possibilities, as JJ and Bennett will talk about those. Again, remind you, you have one job: to help the regulatory reviewer help you and avoid lost opportunities. Of course, you have to remember the analytic implications, like you will find in my papers. The subgroup, for example, they always encourage you to look at subgroups, but you have to be careful. But you will find more details in these papers. Now, this is just my proposal of how you want to structure your thinking and your process in your company to create some kind of benefits communication platform, if you would, or a position. And you have your benefits core team starting early to put a strategy, and you maybe have actually benefits assessment stat plan early on, and all this will have to be within some kind of SOP or working instruction. And then you actually have identify the triggers where you want to revisit this based on either the clinical proof of concept, which will happen very early, to give you a preliminary idea about what to expect. Maybe during the development itself, as you realize more benefits and risks, also in preparation for marketing authorization submission, and if there is any significant post-marketing data or any major change in the management landscape by physician associations. So these can be triggers for you to change your position as a company. And then all this you have to create some kind of benefits governance to be able to organize this, and then align with all the other documents. And that's where it gets dicey sometimes. Now, what is the source of truth? So you have to decide what is the source of truth for you. That's it. So if you have any questions, it's great. Thank you. Okay.
Not following, not following the directions. It's being recorded, so supposed to use that microphone.
Thanks for the hello. Yeah, thanks for the talk. Is there anything in the guidance around how risks should be ascertained, like the frequency of monitoring and that sort of thing? I've just seen, companies like with the same kind of class of drug do it in different ways, and then at the end, when the phase 3 studies come out, it's hard to compare.
No, the ascertainment of risk, actually, the whole process for signal detection and signal management varies in each company, but usually it has for the same kind of broader strokes where you try to figure out whether you have a signal or not. Can you support it by mechanism of action, what's in the literature, what's in the other parts of drug class. So that's for the ascertainment piece. Whether now, once you know whether you have risk or not, potential or identified, now moving whether it's a key or not, that's the other dimension of it. And whether it's key or not, there are so many factors to consider, and the guidance actually lists those to make your life easier. Like, for example, if you have a much higher instance in the background of the patient population, even slight increase will translate to much higher impact on public health. That's just one factor to consider. For the severity, is it reversible or not? Is mitigatable or not? So there are so many factors to consider for the key piece, but that's different than ascertaining whether it is truly a risk or not.
From my perspective, it's not as critical that different companies was assessing similar drugs use the same method as that they come up with a defensible argument and transparent.
Yeah.
So one company might use overkill, they may use a lot of math, and the other says we don't need it. It's obvious if you just look at the data this way. Also, the drugs may be the same, but take one adverse event, throw it on the other side of the zero line, and suddenly you do have to bring in something more intense.
Yeah, that's good.
Thanks. So Tarek gave us a good background for the regulatory context, and also the FDA framework is often referred to as an example of a qualitative assessment. And I think JJ is going to go into a little more detail of how you decide or what things to keep in mind as which direction you go to think about whether you can do more qualitative or semi-structured approach. And Tarek, thank you very much. Are there any other questions or clarifying before you're going to hear a little bit more about some of the things that Tarek touched on in the next couple of talks. Okay.
Yeah, maybe you could help me. Thank you, Alicia. What can I do without you? Anyway, hello everyone. I know it's nice outside, so I really appreciate you being here. But what I can tell you is, without being arrogant, you will have a advantage of knowing more about this. We have a great expert, Tarek, lay out the foundation from the regulatory perspective, and Bennett has been in this field for a long time. Now I will be speaking about method mainly, but before there, just a little bit perspective. This is the 11th year of this workshop done. So I started from the beginning. I remember 2013, it was in Montreal. That's one thing. So this is the 11th one. So always face-to-face, except during COVID. Another one, does anyone of you know that we almost did ICP here, but canceled? 2017, if I'm not mistaken. They were not ready, so we moved to Montreal again. So I'm happy to finally be here. I've been enjoying Halifax a little bit. Hopefully after that you could do it as well. So methodology, I don't want you to be, how do I say, daunted. No, because there's so many methods. So my approach to sharing this with you are two things. Number one, I've been involved in this field for a long time, even maybe one of the first papers published on this one, 2003. And number two, Sanofi may be one of the few companies who do this routinely, deliberately for everything. We have around 3,000 products in the market, development, and so on and so forth. We don't do all of that, but we call it prioritization, which means all product in the development, we do structured benefit risk evaluation. And product in the market that need to have special attention. So annually, we do around 150 per year. One thing I want to do right off the bat is not all benefit risk evaluation is quantitative in nature. In fact, the majority are not. So that's why I'm glad Tarek started the discussion, quantify or qualitative. So I will touch on that earlier. I like the question. It might be too early, right? But in Sanofi, we do it starting 2B. Yes, oftentimes we don't know the whole picture, but the planning is there. And it's a fluid situation. We follow the same framework. We adapt the model if need be as we get more data. So without further ado, it is based on those two principles that I share with you, uh, uh, based on what I know in this field and based on Sanofi practice. And given that this is method, I'm not going to touch everything. I will be on high level in general, except for one, which is for MCDA. Everybody's talking about MCDA, so we will dig deeper. So bear with me on that one. Three things. Maya Angelou said, if you don't know where you come from, you don't know where you're going. So history is important. And I'll bring that briefly. And if you, from US, Yogi Berra said, if you don't know where you're going, you will end up nowhere. So hopefully by knowing where we come from in this field, you see the difference in terms of methods, policies, and philosophies. And then before we go gung-ho into quantitative aspect, we will go into the structured first. What does it mean to do benefit risk evaluation in the structured way? And Tarek had mentioned that because the component in ICH of what are the components of this, how we make a decision about the context, that is part of framework. So we will dig deeper on that. This is maybe the classic example of benefit risk evaluation. Tarek has a more beautiful example in terms of fixing AC, crazy or not, right? So this one is basically, we know aspirin is good for coronary events. The main benefit is fear of heart attacks, but at the same time, the main risk is bleeding. Classic question is, do you take it or not take it? And the answers from the regulators, drug companies, patients may be different. Two different patients may have different answer, but that's the nature of this. And let's go a little bit more complex. Belatacept is immunosuppressant for patient after kidney transplantation to prevent rejection. The drug is approved. But as you see, we're no longer talking about one benefit and one risk, but we have 5 benefits and 6 risks. Okay, just a teaser. You could see that which one is this? Anyway, you could see that belatacept is favorable on everything, on death, graft loss, or rejection. Well, except rejection episode, everything is comparable. So this is another situation you're facing, that people are facing. What is the decision? Again, the answer may be different between regulators and pharma companies and patients. So with that in mind, that these two classic examples, simple and complex, the question when I joined the company 25 years ago, joined industry, is what do we do to combine the benefit and risk, to integrate and make a decision, take into account everything? What do we do? Tarek mentioned SIOMS 12. I mentioned that, I was involved. But before that, this is the first benefit risk document from the regulators, guidance, 98. Now, I will let you read it. But the bottom line is, there's nothing. It is frustrating that there is no defined tested algorithm or summary metric that combines benefit and its data to make a decision. Yes. And I feel the frustration at that time. I was a young person, in industry. I said, what do we do? So myself and my team tried to offer something to quantify, what could we do? So I'm not being self-promoting here, but reading this is part of the history. You will see this is useless at this point. I mean, so many things that are no longer relevant, but at least the thought process is there. There are 2 papers. This is the theory, and there is another example. So, I would suggest if you want to know more, you do it. Another thing is, this paper actually triggered the shift, because I was invited by the FDA at the time, Office of Safety and Epidemiology, or Office of Drug Safety. The name changed all the time. So presented this, they like it. Until this is what happened. I'm not saying that I'm responsible for this, but at least I have a discussion. The paper is published 2003. I was invited by the FDA 2005, start presenting this in ICPI and everywhere. 2009, the FDA started
a project exactly on this. And not only that, actually, Bennett started, this one is Benefits Action Team. I'll dig deeper on that. This is a public-private partnership in the US on this topic. I remember Bennett called me, do you want to participate? I don't know why I didn't at the time, but we have an expert here, Bennett, on this point. Very useful framework, and we'll dig deeper on that. EMA and the FDA, interestingly enough, they started the same thing 2009 until 2012 on this benefits evaluation. And then after that, this is IME Protect, Tarek mentioned Protect. It's a public-private partnership in Europe on benefit risk, part of it of benefit risk. I was invited again, and this time I said yes. So most of my presentation today will be based on this project, because in this project, we test so many methods. So I will present based on that experience. Now, so that paper was published 2003. That SIOM, say, frustrating SIOM is 98. These things was happening until 2015, 14. The shift toward a more structured benefits evaluation, where we do benefits evaluation using frameworks. So that's the second in the agenda I mentioned to you. Let's see what the frameworks here mean. Oftentimes, I'm sure all of you, at one point, people come to you, I want to do this benefits evaluation. I want to do quantitative right away. That's a wrong approach. Before you decide quantitative or qualitative, you have to see the whole picture first. So let's go into the structured benefits evaluation. I'll start with BRAD, Benefits Action Team. Similar to any research projects, the first thing you need to understand is the decision context. What drug you're talking about, what is the indication, what type of patients, and so on and so forth. I'm not going to belabor because my friend Tarek mentioned everything. What does it mean? You see how we really click with each other, so I don't have to talk too much about this here. Do we need to consider everything? Well, maybe not. What type of things that need to consider? So everything will have to be here. This is based on this framework. Once you clear, then you identify the outcomes. What are the benefits? What are the risks? Now, I will explain later on. This is not as easy as it looks, because there are different principles on this, but you have to be clear from the beginning. To your question, at one point during 2B, we may not have enough the complete picture. We may not have enough even the data. Well, we have no data sometimes. I want, for example, let's say HAB1C over there, right? As part of it. We don't have the data, but okay, when the data come in, we could include that. So, and the next part, once you identify the outcome, is what is the data sources? People think this is easy. No. In my experience, this is real experience, one of the outcome is weight change. I'll explain to you later. But we have many trials. We have 60 trials on weight change with different follow-up. That's one example. Do you want to include only trials with 1 year follow-up at least, 2 years, or everything? Where sometimes the trial is only 3 months, stop. It makes a big difference. That will have to be defined at the time. And then after that, the customize. So you get the data, identify, and so on and so forth. You can start the evaluation, and the evaluation may start actually, may stop. How do I do this? I want to have point. So we could stop here. We could stop here. This is in relation to what Tarek mentioned. You don't have to go to quantify everything. You could stop here. Now, if you cannot make a decision over here, then you go into assess the outcome importance. Because one outcome more important than the other. And then, and so on and so forth. So just, this is a flavor of what BREAD framework looks like. Now, this one is ProActURL. On a personal basis, not my favorite. I think BREAD is simpler and makes more sense. But in principle, all the components are here. The problem is similar to the decision context. and then the objective is part of the decision context as well. All of these are part of this decision context, within breadth. But such thing exists. The publications over there we test in Protect, and both of them work. And last but not least, but there's another one, but I'll present only this one. This is also the framework as a result from the Protect project. Again, the step-by-step approach of how to do benefit risk evaluation. Similar, you do the planning, evidence gathering, data preparation, the analysis itself, exploration, including sensitivity analysis, and then conclusion and dissemination. Now, the question from you is how these frameworks compare to each other in the context of the regulatory requirement. Now, this is the summary of it. If you see FDA guidance, it's around the decision context, relevant information, data, weighty information if necessary. Now, all of these are both in ProAcQRL and breadth. So my suggestion to you is, if you want to implement this in whatever company or institution, you could choose either of these. Well, again, in Sanofi, we're using breadth. So that is the structured frameworks in benefit risk evaluation. Now we go into the toughest part of my presentation, which is the method itself. Because as you can see, all of this framework doesn't necessarily explain the methods or doesn't necessarily marry to certain methods. You could use whatever methods to quantify, but this one is more on the foundation of how to do the evaluation. So now let's get to it. Let's get into this final part of my presentation, which is the methods. And I will go slower. How am I doing so far? Too fast? Too slow? Okay, good. So, you know the history a little bit, just to recap. You know that before you go crazy into quantitative methods, learn about structured approach, the framework first to see the whole picture. And then once you understand that, then you go into the method. Now, how do I do this? You do it better, Bennett. Anyway, these, right back. Okay, thank you. This is the result, one of the publications from the Protect project that I was involved in. So you could read the paper. This is a very good paper, actually, explaining the detail of 47 methods and approaches for benefit risk evaluation. Not all of them are quantitative. It could be qualitative and quantitative. The three structured approach that I mentioned early, that's actually considered here as part of the qualitative part. Descriptives, you see, breadth, ProAcQRL is all here. There are 4 categories, the framework, metric indices, and the framework, there's quantitative as well. So again, this is just, there are 47 methods if you're interested in. Not all of them are practical, trust me. That's the reason why what I am explaining right now is the most practical and one of the most commonly used by not only regulators, but also by pharmaceutical industry, by Sanofi for sure. And we will deal with 3 things. Number 1 is the quantitative frameworks, MCDA, SMAA. Tarek presentation said the FDA start using it 2021, right? The publication. Anytime, I think Bennett remember, anytime we talk to Francisco Pignati about preference studies, he kept talking about MCDA instead of DCE. Anyway, so we'll touch on that. Again, not because it's the best, but because it might be simpler than others and the most commonly used. And then we will go into utility survey techniques. You will see the difference. And we will deal with the discrete choice experiment methods. And the last one, because all of you are epidemiologists, I'll explain to you a little bit more on impact numbers, which is, Tarek mentioned it as well, is maybe the closest to us epidemiologists because the concept is very epidemiologic in nature. All right, so let's start with, everybody ready? Go to quantitative framework. I use this as an example, so just, disclaimer, we're not trying to change the decision. The drug is not on the market, by the way, so it's a good choice. MCDA, multi-criteria decision analysis. Before we start anything, these are the principle. The three ingredients, and number one is benefit is evidence data. As an epidemiologist, this is exactly the incidence, the prevalence, the risk ratio that you get from real evidence studies or from the trials. You get that. That's the first ingredient. The second one is the value function. Your question is, what does it mean? Okay, I'll give you a teaser. MCDA, in essence, is combining different benefit and different risk into one value. For you to have a composite measures of different variables with different unit, you have to have a unifying unit. That's the value for that attribute. You have weight, which is in kilogram. You have blood pressure, which is in different scale, and you have HbA1c milligram, and so on and so on per deciliter, right? So different type of unit. You need to combine that. And then the last piece, last but not least, is the preference weight or the relative importance, because HbA1c compared to, how do I say, let's say cancer, as there is, right? Many people may have different things. For people said, okay, I just want to improve HbA1c. So that is the third component, how you weight different benefit and different risk into account. All right, so recap, three principles. You get the data, you have the value function, we'll get to that. And the third one is the weight itself. The principle. Now let's go to the step by step. Now, again, you can read the reference. If you notice, this step is similar to breadth, similar to PREQ-RL. Before you do anything here, because MCDA is a framework, with the quantitative flavor. The first thing you need to do is you need to select benefits and the risk. If you remember in the breadth framework, this is part of the framework, which is the second step, the decision context first, right? But okay, we know the context. Now we decide what the benefit and the risk. Identify the data source and get the data. I don't need to repeat. This is very important because people may pick and choose the data source they like. Oh, I know the result from the 1-year follow-up is really good. So let's exclude only 3-month or 6-month follow-up. You cannot do that. You will have to make a decision upfront. Yes. Actually, for both. Yeah. So well, when you try to do benefit risk evaluation, of course you're dealing with your product, right? Now, within that context, there will be a step when you make a comparison, what type of comparison you want to take. Now, in my example later on, you will see it's placebo, but that decision will have to be made not only by myself, but by different stakeholders within the company. So a very good question. Before we even decide this, you need to decide, again, what drug you're talking about, for what type of patients, for what purpose. Is it regulatory purposes? Is it payers? Is it patient? Is it internal decision making? Because that will dictate, the next steps. And when you're talking about the benefit and the risk, you will have to talk about the benefit and risk with both drugs in mind, the drug and the comparison group. Correct. But at the same time, this is a very good point, important one. Even placebo group, they will have adverse events, right? They will have adverse events. So you will have to see everything relative to that. So when you make a decision about, okay, this is the event here, you have to put that into perspective.
Also, would that place there?
Oh, absolutely. That you may want to consider that. In fact, in our project, you will see the example. Rimonabant is a weight loss drug. We have placebo, we have methotrexate, we have other drugs as well that are already in the market. So all kind of comparison could be done, and you will have to be clear and upfront to what comparison group you are trying to do over here, whether or not placebo and so on and so forth. You will have to be clear on that. There's no right or wrong in different contexts. Alicia, yeah.
Those are great questions. It's kind of the decision context that JJ was mentioning. Those are critical decisions to make up front, is what your comparator is going to be. And when you're talking about benefits and risks, when I've been working with different companies that aren't from, or different groups of people, they want to talk about the attributes of what the positives and negatives were from their particular drug. But you do have to put it into context and think that's the, for me, the hardest part is trying to come up, what are the key benefits and what are the key risks and how are you defining them? So it's not just your own product. Yeah, and Bennett wants to probably.
If you're comparing to placebo, you might be able to get away with just the adverse events associated with your drug. But if you're dealing with an active comparator, you need the union of the adverse events associated with both. Think about surgery versus arthroscopic surgery. In surgery, you're going to have anesthesia. You're not going to have anywhere near the same anesthesia issues with arthroscopy. But if you're going to compare them, I'm going to have all the adverse events associated with surgery and all the adverse events associated with arthroscopy put together. So I show one treatment prevents them and the other treatment causes them. And then it becomes a decision context. If I'm trying to make a decision, say a go, no-go decision, I want to compare with what I think is going to be on the market at the time of launch or shortly after. If I'm doing a regulatory decision and it's appropriate to use a placebo comparator, then I could do benefit risk against the placebo, even though the regulators in the back of their head are going to say, well, what about the other drugs on the market? And usually what happens during an advisory committee meeting is they say, well, we're not supposed to do this, but if I compare.
Yeah. No, this is the beautiful example of what's going on when we do benefit risk evaluation. This is the key points of hearing, listening to everybody, and make a decision together.
It's a bit difficult for him to even convince the governing body what is the benefit of this at the end of the day.
Yeah.
I think it's important for us to highlight that so that
Yeah.
it will be very clear.
And
Because
I
the
feel
trial you are doing, you are using placebo, but there are other competitors in the market. Maybe they've entered the market before you, and your own is coming. And I don't know what you are comparing to, or because.
Correct. No, it's
Cladribine example I
it's
mentioned,
good.
you can look it up. It actually compares the cladribine to many other drugs in the same class beyond the on placebo. So that's a good example.
No. Again, for the simplicity of the method, I use placebo. You will see, but in practice, and if you read the report, I'll give you the reference on the example. We use active drugs on the market at the time, and it doesn't always have to be head-to-head comparison in the trial. You could just get from the other data. There are techniques to connect it. Okay. So anyway, keep the question coming. This is the point. This is not just me sharing, because it's very important that we understand it. So anyway, we discussed this. So this is where the value function is, to unite everything. So we apply the score and then the weight, because MCDA itself is in nature is quantifying it, calculate the score, and do sensitivity analysis. Let's do it one by one. You see, now this is exactly what we have been discussing. There's so many blood and tears and frustration to come up into this list. All kind of things. Many things, and Bennett actually will discuss more about the principle of this, because, for example, you cannot repeat two similar variable or benefit into this. Like, for example, we have over here cardiovascular death, and you cannot put like death, because it's, there will be double counting over there. That's the one principle that we didn't mention. But again, all of these will have to be relevant. We'll have to have the context, behind it. Whether or not this is okay for placebo or for active treatment, you need to have a reason behind this, and you record it. By the time we have more data, we have this, and if you change this, make a reason why we change it. So it's not just fishing expedition to make sure that your drug looks good. You have to be objective to begin with. So assume with all the trial and tribulation, we come into this. We have two benefits, which is weight loss and cholesterol change. And then we have three risks, again, for simplicity: cardiovascular, death, psychiatric disorders, and this. I was there in 2007 when the drug was rejected by the FDA. And this is the reason. I think many of you remember that. Okay, so yes.
How easy it was to define these outcomes? Like, how did you come up with how you did definition?
It's, well, the definition itself, when we did the analysis, this is a little bit late. So we rely on what has been observed in the trial. You know, ideally, we want something different. We want to measure the BMI itself, not the percent weight change, for example. So to the definition, we have to rely what has been done. That's number one. Number two, we have to make a decision which of the benefit it is the most relevant to the evaluation, because there are many. Not only cholesterol, but also they have blood glucose benefit, which is better, and so on and so forth. And I will give you the more complete one, and there are many over here too. But again, for simplicity, we keep like that. I hope that answers your question. So we're forced to use what is available, for sure, but at the same time, discussion between development, physician, we invite patients as well, my team, epidemiology, safety, regulatory, within Sanofi, you know, we discuss what needs to be done. Now, within the PROTAC, we try to mimic everybody with different perspective as well in making this decision. Absolutely. This is too late. I mean, again, keep in mind, when rimonabant is 2007, there is no such thing as structured or quantitative. This is done later, right? So anyway, we got this. I cannot repeat, you know, I don't want to sound like broken record. This is key because, you know, people may pick and choose the follow-up. People may pick and choose the type of trial, the population. You have to make a decision based on the context. Let's assume we make a decision. This is 1 year follow-up on, there's no diversity at that time. You know, I wish we had that, but based on, this is the best data we could get. And you see over here, this is the, in that triangle, this is the data. Benefit is evidence. Forget about value function, forget about, you know, score, forget about weight, just pure epidemiology data. You have the incidence for placebo and for rimonabant. This is what within the framework, customize it. Don't go fancy quantitative. Just see, let's see the risk difference between placebo and rimonabant. We call it forest plot. You see, for cholesterol, rimonabant is better improvement in cholesterol. The same thing, better improvement in weight loss, more weight loss. At the same time, more cardiovascular death. Again, this is risk difference, not risk ratio. More depression, and more diarrhea. This is a classic example of how we do it in Sanofi. Again, we do it for around 150 products per year. Up to this point, people will gather, okay, can you make a decision now? Is it clear that one drug is better than the other? And again, there's no right or wrong. The key is you need to be inclusive and try to make a decision together. Because if people, okay, let me ask you based on this, you know that rimonabant is beneficial from the benefit side. Better cholesterol, bigger HDL increase, and then more weight loss, but higher risk of these three events. Which one is better? Again, no right or wrong, and you must take position. Who think that rimonabant is better, taking into account the benefit and risk? Again, forget about weighting quantitative. This is, I should say in Sanofi, 80%, 80 to 90% stop here. You don't need to go fancy. That's the principle that I want to make sure you understand. Who thinks that rimonabant is better than placebo? Okay, good. The same question, which one is better? Nobody? Okay. Which one placebo is better? Okay, 1, 2, 3, 4, 5. Okay. And it is okay for you to say, well, I cannot make a decision. Which one think that way? I cannot make a decision based on this. Okay, the rest. Good. Everybody answer in general. So again, there's no right or answer. Right? The key, the most difficult part is how as a company, as a team, you proceed and everybody is happy. Right? Okay, for the sake of that one, that's why I want to visit my friend graph on quantify or not to quantify. This is one of the, we publish this. So please read, and all the meticulous information, come to this graph. What we have done so far in this example is basically, okay, we get the data. You see it over here? ProAQL, breadth. We said it in this paper. ITC is the indirect comparison when you compare one drug with the other, but not from the same trial. So you get all the data and everything. You do the plot, classify evidence, benefit, and risk. You have benefit and risk, favorable, unfavorable. The primary author is European. That's why we use European words. And present data on key. That's what we have done. We get the data and we get forest plot. And here it is. We are here. Is one alternative clearly best? If you think, yeah, okay, either placebo or rimonabant is better, then that's it. Forget about the fancy stuff, quantitative. We stop here. That's what we call it qualitative. So the term qualitative doesn't mean it's all about feeling without nothing. Of course, we have the data, which you have to estimate quantitatively. Incidence preference is quantitative, right? So the term qualitative in benefit risk evaluation is that you don't quantify the weight or the relative importance of the benefit and the risk. That is the main first thing that even Sanofi people got wrong. I need to correct it. And I'm at Sanofi. People keep still thinking that, what we are doing up to here, because we are using data, they said semi-quantitative. I said, no, you should talk to Steve. Because Steve still think that way. Steve used to be at Sanofi leading the team. But anyway, and you can see over here, if people cannot make a decision, then you could do the next step. You could go MCDA and so again, MCDA again. Again, I'm not going to belabor. There are different types. There's semi-quantitative, there's quantitative. There are differences in the opinion. Which one is semi-quantitative, which one is full quantitative. It's okay, as long as we understand that up to here is all qualitative. Yes. Correct. In Sanofi, 80-90%, that's it. We make a decision, it's clear. Kill the drug during the development. That's the term. That's why people hate me, because I said, okay, you need to stop this. I mean, at least we have an opinion, right? We have another piece of information to make a decision. Okay, for the sake of our exercise, let's assume we cannot make a decision, and that's where the value function come into play. What I mean by that is, look, we have what? We have HDL, which is milligram per deciliter. We have depression, which is percent or per thousand, death per thousand, and we have weight change, which is kilogram. We cannot combine all of those because those are different units unless we have one universal value, but there should be a function to change the value. And this is what it is. When I was younger, I love statistic mathematical model. So I use Weibull, Poisson, all kind of fancy stuff to transfer the value. But you know what? Now I get older, doesn't make any difference in the end. But there is one thing that is key that you cannot ignore, which is whether or not the thing is good or bad, because you need to have different direction, the same direction. My point is, if you're talking about weight loss or HDL value, which is good, then you have to transfer this into something good. So if you have high HDL, then the value should be good, should be big. That's why this is a positive gradient. That's the only thing. Either increase, either I forgot the math term, either increasing gradient or negative gradient. That's all you need to do. So in this real data, the minimum is based on the minimum value in the data. The maximum is based on the maximum value from the data. And then the scale from 0 to 100, that's it. This is natural. You don't go fancy. And then you draw the line. And in Rimonabant, the value of the HDL is 2.12. Okay, put here 85. Bingo. Simple. This is the good guy. And the gradient simply based on empirical value from the trial. Depression, the same thing. You could see, you cannot see very clearly, but this is the minimum incidence of the prevalence in the trial. Remember, each trial, we have 50, 60 trial, you have different incidence of depression. So you get the minimum and the maximum. And then the scale from 0 to 100, but make it negative gradient. Why? The higher depression, the worse your drug is. So the score must be lower as it increases. So that's it. Forget about fancy model. Just use linear positive gradient and negative gradient, and you get the number. Now, this is the score. Yes. That's the fancy stuff. If you from a trial, you have confidence interval, right? Then the value over here, you could use that. So in fact, MCDA score could use confidence interval, but I'm not going to touch into that. There are techniques to do that. In fact, actually, that is the last topic I want to discuss today, which is the simulation methods. But I don't want to go too fast. This is cozy. We talk like very closely, very slowly. So anyway, we got the score, right? So we translate the value to score. Now the last one, not the last one, the next one is the weight. That this is where the patient preference, and Bennett will talk more. This is the third internet graph. We get the data, we get the value function, this one, the score, and now we ask people which one is more important. So for depression from 0 to 100, what's the score? This is where the opinion from patients, physicians, regulators may be different. Now, in this exercise, we actually, in my team at the time in Protect, we have everybody. So for the sake of exercise, we ask people. So these are the answer for, the 2 benefit and the 2 risk. And this is the overall. Yes. Yes, I got you saying. So that is the very reason why when we deal with this, some people say, don't ask the patient because they don't know, maybe. Ask the physicians. But the flip coin is, what do they know, the physician? It's the patient. Whatever they said is right. Because, for one person, depression is maybe much less important than I want to lose weight. You don't know how depression because of the drug, how bad I feel is. So it's an ongoing discussion how we could do that. But that's why patient preference project, where Bennett, everybody here involved, is we want to bring patient into the table more. Exactly. Absolutely. So again, feel free to add. I add this from this again for the sake of example how it's done. But yes,
and we
it's not
have
easy.
more coming after the break. Bennett's got some
On this, yeah,
examples.
Bennett will dig much deeper on that.
Data. clinical data, often a regulator will make a decision for a population as a whole. They will take a stab at males versus female, old versus young, but often the decision is made on the population as a whole. Preferences will be heterogeneous and will show a distribution, but usually you will think of them as an amalgam. If you want, or if there's a need like before and after menopause, big difference in thoughts about estrogen replacement therapy. There it is definitely worth partitioning your population and looking at the preferences differently, especially because postmenopausal women are the ones who are doing it or feeling the risks, and premenopausal are thinking about it theoretically for their future. It's very context specific, but you could go crazy with it. You could say, what are the differences by region, by culture, by gender, by age, by every subgroup you can think of, and you end up becoming paralyzed.
So anyway, we ask people what the weight is. Okay, there's some tweak. Again, I don't want to talk too much detail, but we need to normalize it. Simply, this is for interpretation purposes. So the way you do it is, so this is for this one, this is 73 divided by this plus this, because this is first level overall. And for here is 70 divided by, for race, right? This divided by this plus this. And for race, you know my point. Normalize it, simple mathematical model. And then the last part is, what is the score, MCDS score for this? Is basically that normalized weight that we just saw, plus the value that we assign based on the simple approach. And then you add together, you got it here. It so happened that the score between Ramona and Compaya is the same, 66. So in this evaluation, you still cannot make a decision. Although in the end, you know what it means? The green means if the more benefit, the bigger the score, more benefit. Now over here, the bigger the score, less risk. Remember that negative inverse function? That's why we use it. So the fact that rimonabant has lower score into the red one compared to placebo is because they're more dangerous. They have more risk. That's what it is. That's the reason why we need to make sure we have the right function, so the interpretation is consistent. Yeah. That's related to what Tarek mentioned. That's where confidence interval come into account. We did that in the project. You could read in the paper. They had a technique to do that. You know what? In fact, let me finish this, because the next topic, quickly, it's taking into account that uncertainty, not the significance, but uncertainty. Okay. So if you want to dig deeper, the benefit score, these are the individual score of, again, I give you example only 3. But in fact, there are many benefits included, which may not be necessary. But for the example, this is the score based on the data from the trial and also the value that I showed you before, the graph, and based on the weight, right? So this is the component of the risk. You see anything here? Not only that, you could see the difference between the two, which one that make more difference. The main difference between the two drugs is actually on the waist circumference. So rimonabant is much better in the waist circumference part, but the most dangerous in terms of anxiety. Again, if you want to go deeper. Now, going back to your question about, uncertainty, this is the importance of sensitivity analysis, which is the last part. Remember, people give weight to benefit and risk. The weight at this point is around here for benefit, which is around 42% benefit. And this is the score, stay here, 66. Suppose you ask, instead of patient, physicians, the physician thinks, oh, this drug is very good. The benefit side, the weight, put more weight on, let's say the physician put more weight on benefit, instead of 42 is 80. Then the graph will be here. If that's what happens, rimonabant score will be higher. So this graph shows you sensitivity analysis, which means the impact of changing the weight of the benefit. And you could do the same on changing the, the risk as well. But this is just an example of how the change in weight could change the equation. And you could see over here, this is not very sensitive, right? Because it's flat. But when this is cross and it is very steep, it's even more sensitive. Okay. In the interest of time, let me, before I close, do one more time. You see what from this one? This method relies on what? The weight. It depends on the weight you assign to benefit and risk. But there is a method where you could completely avoid the weight. This is closely related to MCDA. This is rarely used. We use in Sanofi once in a while, sometimes, when we cannot make a decision. So this is what we call it stochastic multi-criteria acceptability analysis. This is based on stochastic process. This is similar to Monte Carlo simulation. So basically, here is the step. This is similar to basically MCDA done 1 million times. Exactly like that. That's why I call this the big brother of MCDA, or little brother, maybe. So let's do it one by one. We get the data, the same thing. We get benefit and risk, the data. We get the score, remember that graph, do the thing. And then weight instead of, okay, sorry. The data, we get the clear data. Over here, you get the data randomly from the confidence interval of that variable. Before in MCDA, oh, the data is what, 2% or 2. No, over here you pick randomly from the confidence interval of that variable. Both rimonabant and placebo. Okay. And then the same utility score, you ran the graph, translate the score. The weight, remember, we asked people, what's the weight? In this case, no, you pick randomly from 0 to 100 for that drug, for that event. So let's say 50, 60, same thing. And then you calculate the score, and so on and so forth. And then you will get the score for rimonabant and placebo. That's only the first iteration. You do it 1 million times. Same thing. Pick randomly the data for, or the value, not the value, the data for that variable, that attribute. Get utility score, get the weight randomly. You do it, this is just illustration. Let's say the first round, placebo win. The second round, rimonabant win. And do it 1 million times. This is the real one. This is the real one. In that project, what happened, rimonabant show up as the preference drug 70% and 30% as second drug. Well, of course, because you have only 2 drugs, the flip side is for placebo. And again, I'm a Sanofi guy. I didn't do anything. You know, we have people running this, but this is what happened. So the benefit of this is you completely free yourself from assigning, how do I say, weight, anybody. I mean, this is the machine. Number 2, to a certain degree, you address the uncertainty, the variance, because you pick randomly from the confidence interval. But Sanofi hates it, because what does it mean, the interpretation, which is true? Lack, how do I say, clear medical interpretation, because everything is based on, random process.
You get what I'm saying? Instead of this.
Oh, I see.
Yeah.
Well, that's a good point.
Yeah.
Because when we ask physician, I said 0 to 100.
Yeah, you have different distribution. You have a distribution.
Correct. That's a
Yeah.
good point. We could see based on the answer from the patient, right?
Yeah.
You know the scale, pick
Yeah.
from that.
Yeah.
Very good point. So you
Yeah,
may
yeah.
have different scale. Yeah. We didn't do it, but
Oh,
but
I think is,
something.
yeah, I think is a
something
better
that could be
approach.
done.
Yeah.
Okay, so I think we could stop because the okay, I can do it 5 minutes. Any questions so far? Because again, the reason why I take time on MCDA is I can see more and more that people are using it. It's not easy, I know, but at the same time, what I heard is easier to do even than this one. Any questions so far before I continue? Reza?
Not an out of the box question. Instead of the score that people are giving, if we put the burden of diseases there, so the DALI, do you think what would be the difference? Is it doable, or would it be the same result?
You
or
mean the
different-
score based on the function instead of which score?
of the utility that you develop?
This one?
Yes, yeah.
Okay, okay.
Instead of weighing this way, if we put the burden of diseases, which is still between 0 and 1, or to 100%, it's
Burden of disease where?
DALI.
Oh, DALI. Yes, yes, yes. Okay, okay.
So basically, the DALI certainly will not go to the HDL, but for depression, and if the other one is broken knee, something like this, that could be done. So
It could be done. In fact, DALI, Kali is part of our consideration, because when we did Protect project, we consider all 47 to be tested. We ended up not using it simply because of the lack of data for this particular drug. So yes, in the bigger scheme of things, when you're talking about other areas, the reason why I put MCDA here alone, it doesn't mean that this is the truth, that this is the best, but just to give you a flavor. Yes, I think in the different situation, different method may be more suitable than MCDA. The quality, maybe DCE even better than this. Reza, what did you say?
actually, I'm not an expert, but I try my best. DALI is basically based on the opinion of a large group of people, which has been validated again and again in terms of the trade of life, one to other one. Let's say I have a very bad headache, migraine headache. How many days I'm going to exchange when I'm 80 years old to give it just to get rid of this pain? It's one part, which is the quality of the life. The other one is the extension of the life.
Yeah. So the quality is quality adjusted life years. And okay, sorry. No, I just want to make
Yeah.
sure the
Age
audience.
adjusted life years and quality adjusted life years.
Yes, very good, very good.
Thank you.
Qualities and DALIs have similar underpinnings. When you want to use weights and benefit risk, you actually want to use, in my view, the components that go into the non-mortality component of a DALI. So a DALI has a length of life change and quality of life change or disability. That disability is assessed with some function, a disutility, or the 1 minus utility. That utility is what would go into weights here. You don't want to mix it up with death. You want to keep the weights for death associated with the endpoint for death and the weight for stroke and heart attack and the like separate. Now, if you happen to combine them and do a probabilistic model over the end of life, it'll be very similar to a quality, or if you inverted a DALI. But I always thought that the DALIs take it too far, and I need to go earlier if I'm going to use weights that I can defend and mix it with my clinical data.
I think in the interest of time, the DCE, I think, is very close to Bennett presentation anyway, so we could skip it. And also the last one that I mentioned before. So all of these, Bennett will touch on it, so there's some repetition. And this is NNT and NH. You could read the paper, and this is a very simple topic. But again, the closest to that, I'm sure by reading the slide, which is available, you will have a clear understanding what it means. But again, my objective today, I think at least if we could discuss MCDA ad nauseum, so to speak, and I really like the interaction. And SMA, I think we there. So I know it's break time. So, if you prefer to continue with this.
We can take a poll. We could take a break, or if you want JJ to go through the NNT and NH, we have time. Yeah, that usually take 5-10 more minutes. That's
Okay,
fine.
all right. Well, in that regard, let me go here. So
The coffee's not going anywhere.
The main thing of difference between here and MCDA is in MCDA, you get the data, right? And you ask patients, what is the weight of, different outcomes with has nothing to do with the data itself. It's just the concept. Now in DCE, the patient or the responders will take the value into account too. So in this case, both the weight and the data are taken into account at the same time during the talking with patient or with physician. In MCDA, it's two separate topics. You will see that very clearly when during the example. So again, DC is discrete choice experiment. What we do is in this case is you know what? I'll just go to an example. So you have two options. A is rimonabant, B is placebo. Each option has different criteria or attributes, people call that, which is the benefit and the risk. And each value, each attribute has certain value. Mild improvement, 10 out of, see, this is the data itself. So in MCDA, the data is based on trial. The data is the data. Over here is scenarios. So you change scenarios, in different scenarios, you have to get two treatment, different type of attribute, benefit and risk, and each attribute have different values. And you modify this again and again, different variations. And you ask, if suppose you have weight problem, you want to have weight loss problem, this is drug A, this is drug B, and this is the attribute, this is the characteristic. You got mild improvement for this drug, no change here. So this drug is better for this aspect, but okay, this one is good too. Oh, sorry, but this one is not as good, right? And plus, you have more psychiatric condition and so on and so forth. So the way if I am a patient or a respondent is kind of confusing. That's why I like MCDA more, right? Because you have to take into account all of this together and you answer, the patient or the respondent answer, based on this scenario. Let's say I take drug A for one person, the other one take drug B. And you repeat this many times, the same two treatment, the same attributes, but you vary the values all the time and see how people answer. And then based on those answers, and this is, Bennett maybe will dig deeper, this is where there's a limit to how many combination of this that people could handle, right? Could handle logically, and also how many variation of this. So there are discussion about that. I'm not going to belabor too. But the bottom line is, this is how it is done. And based on the answer, this is similar, simple statistical calculation based on yes answer no. You could use logit probit. In this case, you use probit. You could see the how to say association between the variables and the decision. So what it means really is for weight loss, the higher the weight loss percent, the more people choose A. Of course, right? But the higher the depression, the less people choose A. Of course, it's the bad thing. This is comparable to that graph that I mentioned before, down and up graph. And so you could calculate it, and then you could calculate the value overall, not only the attribute, but overall drug value based on this. And in this case, this is the formula. We're not going to belabor. You could see the rimonabant, the value is around 56 or something, and placebo is slightly low. This is another angle of how to do it, right? Again, there is no, how do I say, universal truth. Okay, use MCDA, use DC. There are pros and cons in between. So this is just a little bit of flavor before my friend Bennett will dig deeper on both ways. Last but not least, I cannot, I originally want to erase this from my slide, but we are epidemiologists. Epidemiology one-on-one, you're talking about, prevalence, incident, burden of disease, and association. These actually also very useful for benefit risk evaluation. But most of this is one-sided in either for benefit alone, risk alone. You cannot combine the two, right? But I thought this is the paper that I wrote in back 2003 mentioned about this a lot. You know, the main basic concept of benefit is evaluation. So there are many things. Attributable risk, everybody knows this is, you know, risk attributed to the treatment, which is basically based on, you know, race difference. And what I want to touch over here is, because this is popular among physicians for some reason, is number needed to treat, or what or call it exposure impact number. Number needed to treat or number needed to harm, which is 1 divided by attributable risk, 1, which is the inverse of risk difference. This is funny because we have a big debate on it. What does NNT means, right? What does NNT mean? Number needed to treat depending, or harm depending on benefit or risk, which is number of people to be treated for 1 additional outcome. This additional, oh my God, is necessary. This is what epidemiology come into play. Why additional? Because rimonabant or placebo have the outcome too. That is the difference, people who understand epidemiology or not. Sometimes people say, okay, to have 1 outcome, not 1 outcome, 1 additional outcome compared to the whatever you're comparing to. Because this is the difference. This is not absolute. So again, just to give you an example of how important it is. So the point is, you know, based on this mathematical alone, yes, everybody will have weight loss, right? Even people not taking the drug, they have weight loss. Some people gain weight, but you will have certain people having weight loss, let's say 5% over a year. The question is, how many people you need to treat, assuming everything is the same, to have additional weight loss in addition to, you know, the base. That's what it is. So that in itself is a benefit risk evaluation, because you know what? You know the benefit of a drug. You don't know the harm, but doing that, you know the benefit. So, but when you're doing benefit risk evaluation combined, you could do simple thing. You could compare NNT and NNH. Let's pause over here. What does it mean NNT is less than NNH? What does it mean NNT is more than NNH? I'll give you an example. Weight loss, NNT, no, forget about the others. This is based on 2 data trial. In general, all the trial, NNT is 4. Influenza, this is NNH, because flu is bad, is 40. Is it a good thing? Is this drug good? The fact that NNT is lower than NNH? Okay, let's do a quick quiz before we have coffee. What is it, a good thing or bad thing? Who say good? Who say bad? Good, at least you answer. Good. Correct.
Here, number of people tenfold smaller than now
Is it a good thing or bad thing? You're very helpful, Bennett, because the way, this is where I think the physician come into play in terms of their perspective. I need to, by the time I get 1 influenza, I got already 10 people having weight loss. That's not a good thing. Because for you to have 1 influenza, you need to treat 40 people. When you treat 40 people, you will have 10 people losing weight. So by the time you come here, you have 10 people lose weight, and only 1 people have influenza. So it's a good thing. Yes. Very good point. I ignore this for this example, but you are right. You could this is maybe not a good example. because you have to take it into account. Good eye. In previous year, actually, I used diarrhea as an example, simply because we don't have complicated here. But I said, I don't want to use that again. That's gonna ew. So, but anyway, very good point. But let's try to figure it at this point. Just assume that the confidence interval is okay. But that's how we interpret it. And of course, and you know, you all of you expert in this. You don't take into account the relative importance between weight loss and this. Because for these people, I don't care if it's like 10 times the value of this, I don't care. I don't want to have friends. So that's why MCDA comes into play, because there is no weighting whatsoever, and there is no combining between benefit and this. This is becomes subjective. So I'll stop there. I think we reached the point when we maximize our time before we have coffee. What do you think, Alicia?
One more minute, and then Bennett will give Carol's talk, who she spent the day yesterday in the plane or an airport trying to get here. She left Raleigh, Durham, and went to DC. And once she arrived at DC, found out that her flight to Halifax was canceled. And then she spent more time trying to rebook her flight to get back to Raleigh, Durham, which if you drive is only 4.5 hours. So she spent all day yesterday trying to get here. So thanks to Bennett for picking this up.
All right.
Sorry. One more thing. I know we have a small group here, and the questions have been fantastic. So please keep that up, because that makes it more interesting and informative for everyone that's here. And there will be evaluations. And since we have such a small N, it will be very appreciative if everyone could complete them about the content. We are trying to think about what's relevant and try to focus on what's important for next year's theme. So thank you.
Well, good afternoon. So I'm Bennett Levitan. I'm from J&J. I'm very familiar with Carol. We've worked together on many projects over the years, and I'm very comfortable presenting some of her work. So some of you may wonder why we get into this field. I could tell you a bit about myself. Benefit risk is a close cousin to decision analysis. And, I do want to do something to help all mankind for all time and to help patients. But ultimately, I just wanted to prove to my mother that I was right. Decision analysis does not work at home, so do not try it there. Okay, so we're going to talk about patient preference methods. So let me start with, make believe you're a regulator. New drug comes to you, say a triptan. Now, triptans are vasoconstrictors. They can cause heart attacks. They can squeeze your coronary vessels, and they're contraindicated in people with cardiovascular risk factors. But those people get migraine too. So suppose somebody gives you a triptan, and it does a great job of reducing pain and allowing people to function even when they have pain. But it has a 1 in 10,000 chance per year of causing a heart attack. Would you approve it? Well, it depends how important a heart attack is compared to relieving pain and allowing you to function. What if it's 1 out of 1,000? What if it's 1 out of 100? Well, at some point, your judgment as a regulator becomes uncomfortable. That's where patient preference studies come in. The patients live with migraine. They know what it's like to have their lives suddenly disrupted twice a month, say, for acute migraine for 1 to 3 days, and suddenly not being able to do their work, or they have to stop their job as a surgeon or whatnot. And they could say how much risk of a heart attack they'd be willing to take. And in fact, in the preference study of 200 adult migraineurs, if you were to completely remove their loss of function, so instead of being stuck in bed, they could go to school and work and so on, they would take a 1 in 500 annual chance of a heart attack, which is huge for a population. But the patients are saying the benefits are equal to the risk. You figure that out from a preference study. We're going to look at here when preference study can be useful, some of the methods, some of the benefit risk, some of the actual methods for preference assessment itself, and some of the current guidance on the field. Okay, so when you have a drug, say a standard of care, your new drug will have some improvement in efficacy and some change in risk. Now you want to be way to the right and you want to be low, but this is where you are. Now, in the patient's mind, there is a certain trade-off. As I improve the efficacy or effectiveness, I will accept more risk. That's what that blue line shows. A preference study assesses that blue line for an individual or a population. Now, if this is the risk you've got, this level, that is the minimum acceptable benefit. If I have this level of risk, I have to have at least this degree of benefit, or I'm not going to bother with the drug, at least according to this simple model. Now you could do it, look at how much more benefit you've got. I actually go all the way out here, so I have this additional slack, you might say. I can afford some heterogeneity in my benefit, and that's one way we essentially do sensitivity analysis. If the confidence interval for the benefit is small compared to that gap, you don't have to worry so much about noise. Now you could do it the other way. If this is my benefit, this is the degree of risk I could take. That's called the maximum acceptable risk. And that's much what I described for the triptans case. If the benefit is preventing all loss of function, I could take up to a 1 in 500 annual chance of MI, of heart attack. The maximum acceptable risk is a common metric in preference world, and we'll show it. And then there's a safety margin if this is actually where you are and your maximum acceptable risk is above that. Okay. There are lots of roles for preference information besides benefit risk. What endpoints do people care about? I can design a trial. What changes in those endpoints are critical? I can come up with effect size. Target product profile, the relative importance of benefits and risks or trade-offs, very similar to benefit risk, but used strategically instead of assessing benefit risk. Then the actual defensible benefit risk argument, maximum acceptable risk, and other different metrics. I could get to the approval, where I bring that information and then think about in reimbursement heterogeneity, or how do different populations change? How do different patients and physicians differ in preferences? Sometimes they're similar, sometimes they're different. Are there distinct subgroups, which means I've got to do this analysis differently for people, say, pre and post menopausal? And then the difference between stakeholders is particularly important post-approval in shared decision-making, where a physician might say value death more than disabling stroke in anticoagulant, but patients do it the other way. Okay, so we're really going to focus on benefit risk and a little bit approval and the TPP concept, but there are many other places where preferences come into play. Okay, now there's the concept of preference-sensitive decisions. Where might you find a preference study of use? Well, those are decisions in which basically the decision will depend on preferences. There are times when it's glaring. Benefits way out there, you got no noticeable risks, you don't have a preference-sensitive decision. Or there's a little bit of risk, but any human being on the planet will look at that and say, sure, you got a little bit more risk, but it's obvious from introspection and your judgment the benefits outweigh risks. Preference-sensitive decisions are really ones where you're going to have multiple options, multiple treatments, and the decision is going to depend on the particular preferences. So it might be that you're going to have multiple options, and one is better on one property, the other is better on another property, and you can't just look at it and say one is better. Or there's a lot of uncertainty. I'll show you this graphically in another slide. Now, just to let you know, this was the original definition that came out of a group in the device world, and then the FDA added, preference sensitivity also depends on whether there's a lot of heterogeneity in the patient's viewpoint, or the patient view differs from healthcare professionals. And references can take you into that. But here's an example. Suppose we have a treatment characterized by two properties, a benefit, which we want large, and a probability of infection risk that we want low. So the ideal is bottom right. Now, if your 5 different devices or drugs look like this, well, obviously C is the best. It has the largest benefit, the lowest risk. You don't need any preference information. You just look at this and you're done, like the qualitative benefit risk. Suppose it looks like this. Well, A is better than E and D. C is better than B, but I cannot choose between C and A because one is better, the C is better in the benefit, A is better in the risk. Shall I start again? Okay, so this is an example of where you need a preference information to choose between A and D, A and C. Now, it might be glaringly obvious if you look at the actual numbers, but that's the concept behind it. Now, here's the uncertainty. Let's have the circle become an oval, the width of which and height of which represents uncertainty in the two properties. Now, suppose C has that big band of uncertainty and B and E have smaller bands. Well, on average, C is better. But B and E might have a lower probability of infection than C because of the overlap in the distributions. And if there are patients who are going to weigh infections very heavily and they're really uncomfortable with uncertainty, B may be a better choice because even though on better, average C is better, C does have that lobe up here with worse risk. So that's another example of where how a decision becomes preference sensitive, in this case, just due to the uncertainty. Okay. In general, when you do benefit risk, like you saw before, we assess the benefits and harms. We have the clinical data or epidemiologic data. We get weights, and then we apply them. Now, some preference methods are sort of really the entire process. Like this thing called, we heard before, multi-criteria decision analysis is an entire decision-making process, only one step of which is assessing preferences in the middle. And something called swing weighting, it's done all the time, and I'm just going to abbreviate it, MCDA. Okay, now some methods will only talk about the weight part. I bring this up just so that you don't sort of see, oh, why MCDA, why is it different than swing weighting? Well, swing weighting technically is a part of MCDA. Some methods will look at one weight at a time, like health state utility, or a method called threshold, which we'll show in a bit. They will get you one weight for one endpoint, then another weight for another endpoint, and on and on. Other methods are going to look at all the endpoints at the same time, like the discrete choice experiment or conjoint analysis JJ showed at the end. And stated preference, which is pretty much the same as conjoint, and discrete choice experiment are about the same, do that. Some methods are called revealed choice. This stuff was stated. I show you something and you tell me what you're going to do. I state it. Sometimes I look at what people actually did and I go back in time and infer the weights. Those are called revealed choices. And you might think, well, revealed choices must be great. They show what really happened. The problem is there's so many confounding variables. Like, where was the drug on the shelf? What did my mother tell me yesterday? Or what happened in the newspaper the day before? So there's so many confounding variables that revealed choices are actually even hard to conduct a revealed choice experiment, much less compare them to stated choice preferences. All right. Now, preference data isn't just the numbers. It actually is thought about on 3 levels. The first level is what matters. So the attributes, how important are, what are the things that matter to patients? And you could often do this qualitatively with interviewing a modest number of patients in a panel or interviews. And there's simple qualitative methods that can get you reliable data, and sometimes it's even publishable. Now, relative importance is how much it matters. So not just that this matters and this matters, but this matters 3 times as much as that. For example, in that migraine example, even though pain is the pathognomonic definition of migraine, loss of function, being stuck in bed, is twice as important as severe pain, because that's actually what the patient's cared about. Here you will have to use quantitative methods. And then there's trade-offs. How much of this is worth how much of that? How much of a heart attack risk is acceptable for how much relief of pain or relief of loss of function? And here you will have to use weights and compare them against one another, and you will need quantitative methods. And probably is obvious, but as you go from top to bottom, you're increasing in complexity and quantitative nature. And what's important is sometimes this is all you need, especially for internal decisions and, say, picking endpoints for a trial. But if you're going to do hardcore benefit risk, you're definitely going to need the stuff on bottom. All right. There are a ton of methods. There are actually about 20 at this point. This was done a couple of years ago by the Medical Device Innovation Consortium. There's a new set of work that came out of the public-private partnership, IMI Prefer, that JJ mentioned. And there are about 20, 22 different methods. In practice, about 5 or 6 are really used. So it's more to let you know that there are a variety of techniques that you'll see. But if you were to work with an expert group, like some of the academic consulting groups, you'll find that there are a small number of methods that get used most of the time. One of them is discrete choice experiments, the one Jay showed. The study object in a discrete choice experiment is the thing that you're going to make a decision about, the bundle of benefits and risks and tolerability issues that is your drug or your device, or in general, the alternative that you're going to choose from, say, where to situate a power plant, what to name a child. You break it down into the different components that make the decision. So efficacy and safety, could be tolerability, which is similar to safety, but it's a different flavor. Dose could be an issue because I might not want to take such large doses. I might not be even able to swallow a large pill. Cost, if you're doing health technology assessment. Once you've broken it down into the ingredients, and these things are, you'll notice the word attribute. The attributes that you use in a preference study are not always the same as the benefits and risks and benefit risk. That's usually because trial endpoints are defined in one manner. They're not necessarily interpretable to patients. They can be extremely broad or extremely narrow. You have to follow a set of rules for these things to be interpretable to patients so that when you do a preference study, you really trust what you get out of it. Eventually, I stop touching the keyboard, you do a choice task, some experiment where you show people information about benefits and risks, tolerability, and the like, and the choices that they state go into a model. Often it's a regression model, and the beta coefficients can be translated into weights. And then out comes the information and the preference weights. And that what's not shown here is then the application of it. In a typical preference survey, you're going to first determine, are you appropriate for this survey? Then informed consent. You'll do a lot of background information, like have you experienced the disease? Have you experienced these side effects? Why is that important? Might be obvious, but say in an Alzheimer's study we did, if you had took care of a family member or a friend with Alzheimer's, you were much more accepting of risks than if you hadn't. Not too hard to imagine why. You need to know that so you can stratify your population. If you have experienced an MI versus not, and you're doing a cardiovascular study. Then you train people. So you saw some tasks before. How do you interpret those graphics, those grids, those probabilities? You can't trust people can interpret a percent of probability off the top of their head. You can't trust people know what an MI means, or they know, but everybody's know is different. So you want to level set. And often I'll say, this is the stroke I'm talking about. If there's a wide diversity, I want to define that stroke. Because even though I'm getting less, I'm solving a smaller problem, I trust the problem. Then you actually do the choice task. You show them questions which have different information about those different benefits and risks, and you see what they say, what their choices are, and that will go into the analysis. And finally, you usually clean up with a couple of demographics and maybe questions about numeracy, how well they understand numbers, what type of risk attitude they have, and so on. That's typical of a preference survey. Okay, now, when I elicit weight, you heard a little of this earlier from JJ. I can't ask everybody about everything. First, this is a diabetes problem where I'm delaying onset of type 1 diabetes. These are the properties that we thought about. I was involved in this. How long until I'm now independent, my child is dependent on insulin? What are the chances that my long-term health complications will be halved? What is the chance of hospitalization due to diabetic ketoacidosis, and so on? There's 6 properties here. If I were making a decision, I'd probably have many other things in my head. What we try to do, especially in the qualitative components, is identify the attributes that are most important to these patients. And you don't always know it. This time until insulin dependence, we didn't include that originally. When we did qualitative research, the parents were telling us, we need time to prepare. And that turned into this, because you could do things like get your kid through school faster before they become dependent on insulin. Or you could do family activities while they're not dependent on insulin. They cared about that. So that became a property that went into the quantitative survey. Now, another aspect of these levels, I can't look at every single continuous value of time till insulin dependence. I'm only going to be able to show people at most 15, 20 tasks before they're exhausted. Because these are tiring surveys, and you'll start getting garbage data or people give up. So I can't ask about every number. We spend a lot of time deciding what are the levels that will tell me enough about the answer that I can apply it to my trial data. And usually we try to capture the real range of what we think is going to happen, as well as make the risks high enough that people would say, no way. Because if you make your risks too low, nobody will ever trade off. You won't learn anything from your preference study. So picking these levels is an important part of designing a survey. That you're not on your own. All of this stuff is done with an expert in preference study work. Even a company like mine, which has people who can do this, we have PhDs in preference work, will work with an expert company because it just takes too much time and expertise, and we don't want to allow our bias to get involved. Once you've defined your attributes and your levels, then you have to make a choice task. Now, this is an example of a choice task from that diabetes question. Yes.
From this slide, by the time you are talking about the attribute and levels, this is from a patient preference study, or what would you consider like the consideration from the caregivers, the physicians, where they could contribute, conceptualize the attribute differently?
The properties can be done the same for both patients and physicians, but then we might define them differently because we'll use the right language. And then we could do the survey both in patients and in physicians.
Right. Yeah. But I'm coming to a situation where, you know, the physicians may always consider the efficacy more importantly, whereas the patient will consider 3 days of nausea not that important. So by the time once you see this result using the same attribute levels, but on the different population, the result could be a little bit different or quite different.
And that's great information. I want to know that. If I have physicians and patients who are disagreeing, I'd rather understand why they disagree. They could look at the same clinical data. A preference study can tell me why they, at least one level deeper, why they disagree. So the one that's most common, I think about a lot, is the atrial fibrillation study we did, where physicians ranked death as worse than disabling stroke, and patients ranked disabling stroke as worse than death. Now, as it happens, it often doesn't make a difference because they go in the same direction. But if they went in opposite directions, you'd have patients and physicians arguing back and forth. A preference study lets you disentangle what's behind their thinking. It won't solve the problem. It's not going to make one group agree with the other, but you'll understand what's behind the difference, and then puts you in the position of asking, well, whose preferences should we use? The person who prescribes the medication or the person whose life is on the line when they take the medication? You can see my bias. I would rather use the person whose life is on the line. Yes.
You don't know why, right? You don't know why physician is picking this and why patient is going in that direction. It's just quantitative. You don't know the reason behind their decision.
If you do a standard preference study, no. If you do the qualitative research before, you give yourself some opportunity to start picking up that information. And sometimes it's rare, but you can go back to the same population or a subset of it that did it and do a sanity check. You show the results of the preference study and you ask why. Often when we design these, we go through a pre-test phase. We've designed the survey and we have an individual read through it and answer it, but also give us their thinking. It's not going to tell us the information on the population level, but it'll give us some clue. And then we could design some additional questions to add to poke around. But in general, you won't have the population level rationale.
In your analysis, do you mix the data together? What ratio of physician, what ratio of patient do you consider?
Generally, we will use a couple of hundred people in the discrete choice experiment, like the one we'll show you here. Other methods, you can use a smaller number of people, something called multidimensional thresholding. You could get guy with 50 to 100.
Don't mix the patient. You keep the patient separate from the
Yes, they're
prescriber
totally separate studies.
results.
One in patients, one in physicians.
And I think you have an example. I think there might be an example where the
And
differences.
Yeah, not in this one, but if I were doing, say, a heart attack as a attribute, in the patient survey, I probably will just say heart attack. But for a physician, that's not enough. They often need to know a little more information about the nature of the MI. So the troponin information or enzyme information. So I will give them some details in it. And technically, there is a little bit of difference. Probably the patient is wide range of MIs and the physician is more clustered on a definition. But as long as I understand that, I know how to interpret the results. And yes, they are done separately. Okay, so a choice task is some sort of experiment where I show people, in this case, some information and I ask what you would do. Now, in this case, there's 3 different profiles. I can give you treatment A, treatment B, or I could just watch you and watch the patient without any treatment. Now, this is a hard task to look at. It's kind of a complicated one if you didn't go through the training. By the time you see one of these, you've gone through about 20 minutes of the survey, which would teach you each of these different attributes and how to read these. But to give you a general sense, treatment A and monitoring has 6 years until it'll depend on insulin, which is great. Now, treatment D needs 4.5, not as good. And if I only watched my patient, watch my child, it would take 2 years. Okay, so obviously I prefer this one. Reduce the chance of long-term health consequences. Obviously, B is better. You go through this, and eventually you'll say, you know, I prefer A or B. The reason we had monitoring only, because we were concerned that sometimes people would say, I don't want any treatment. I'm just going to watch these. And we're done. People will opt out. That's really important to the FDA. They've actually said in a recent survey that we wanted you to have done an opt out because you're sort of forcing people to pick between A and B, but you're not learning anything whether a patient would say, I don't want any of this. That's what's going on here. Now, this is a particular set of benefits and risks. As JJ was saying, when you do this experiment, you show people multiple combinations of benefits and risks, and you ask them to choose A, B, or C. And the regression basically says, I'm going to use the inputs are these different variables, the covariates, and I regress on the probability of choosing each of the 3 alternatives. And the beta coefficients are the importance of each row. Results could look like this. Now, down here are the 6 different attributes that went into it and the different levels. And here is a normalized weight. Anything on the positive side is good, I want. Anything on the negative side, is bad, I don't want. When you see a big range from top to bottom, it's important. When you see a small range, it's unimportant. In fact, there's no meaningful difference between those. Basically, it's saying, the patients who did this saw no meaningful difference between no chance of diabetic ketoacidosis and a 1% chance, but a very big difference between 1% and 4 or 5. And for whatever reason, 4 and 5 had a very big difference, much about even, on par with the difference between 1 and 4. Now, if I were reviewing this, I would wonder about that. How do you use this information? Well, you could begin to look at it this way. I can look at, well, okay, that's not what's important. That's what's important. This is really important. But the way you really begin to use this is compare. There's really no absolute scale for preference. The value of something is based on what you're willing to give up for it. So if I go from 2.5 additional years, go to 6 additional years until insulin dependent. So that's a benefit. You see that arrow? That's the change in this preference scale that it's worth. Now I'm going to move that change right over here. That arrow is the same length of this arrow, but it's in the opposite direction because I'm changing the chance of serious infection from 3.3 to 0%. That change is just as important as that change. In other words, from a preference point of view, the difference between 2.5 and 6 years, additional years insulin dependence is just as important as the difference between 0 and 3.3% chance of serious infection. That's the basis of a lot of the trade-off calculations or maximum acceptable risk. And in fact, if we go here, if I say going from 6 additional months to 2.5, that benefit, the maximum acceptable risk of serious infection is 3.3%. I'm the regulator, go back to the beginning. You gave me this drug that would delay the onset of insulin dependence for type 1 patients, but you said this chance of infection is 10%, way too high. Patients wouldn't accept it. 1%, okay, I will accept it, roughly. You have to be careful with these. You have to take into account the confidence intervals here. The maximum acceptable risk will depend completely on what benefit you're talking about. So going from 2.5 to 4 years is a much smaller benefit. The maximum acceptable risk is smaller. 6 months to 4 years, much larger, 4.5. Now, this concept of maximum acceptable risk is used all the time. Here's one from progression-free survival in a cancer study, chronic lymphogenous leukemia. We're looking at how many months of progression-free survival, which is the combination of progression of your cancer and death, are worth various side effects. So if I go have mild to moderate diarrhea, all right, it would be worth about 6 months. No, if I have serious diarrhea, it's 22 months. In other words, it's possible that your drug benefit outweighs your risk, even if it causes severe diarrhea, but you got to improve my progression-free survival by a lot, 2 years or more. Now, you can play games by adding, well, my drug has a 4% chance of this, and it only causes mild diarrhea, and it has a 15% chance of infection, and you can add them all together mathematically and say, well, how much PFS can offset that? There are all sorts of mathematical tricks you can do to say, well, what's it like for my particular drug? Sometimes it's better, though, to go into use a different method, not the preference method, but for analysis, and it's called choice share. Choice share is very similar to market share. It's basically asking if I have 2 or any number of alternative treatments, what proportion of my patients would pick one versus the other? In a sense, it's running your preference study backwards. Instead of showing properties and asking, did you pick this one or that one? I'm saying, did you pick this one or that one, and show me the properties? So here are 2 drugs, A and B, and they differ in certain ways. And A is clearly preferred by people. 99.1% of people would pick it based on the preference calculation run backwards. Now, suppose I change the medicine B in one way. Instead of having moderate to severe palmar-plantar syndrome, I only have mild. Everything else is the same. Well, that's an improvement. More people pick it. If I change the number of months to maintain daily function from 3 to 9, more people pick it. What's going on here is I could create make-believe drugs, or I can make a scenario for what I think my drug will be, or what I think my competitor drugs will be. Or I don't know what my drug will be. I have a whole bunch of different alternatives for the way my drug will go. And I could calculate how the patients will pick from them. Very similar to market share, except it's only based on benefits and risks, not all the other issues that go into market share, like advertising and cost. This is actually a useful approach because, well, HTA people find this much more interpretable than the benefit risk calculation. And the FDA actually devices used this in approving a device a couple of years back. So that is discrete choice experiments or conjoint analysis. I show 2 or more profiles and I ask which you prefer. Now here's the second technique we're going to show called the threshold technique. Let me start it conceptually. I'm going to show you 2 treatments and ask which you prefer. They're only going to be based on 2 properties, 1 benefit and 1 risk. Now suppose you pick the alternative, the one on the right. Okay, I want to find out where the threshold is. I'm going to make the one on the right worse. And I'll show it to you again. Are you going to pick the one on the right again? No, now I'm on the one on the left. Okay, so now I might make the one on the right better. And eventually, I'm ping-ponging back and forth, and I find where you're essentially indifferent between the choices. What that can give me is a point of indifference. And with a little bit of math, I can come up with a weight. So I'll have a standard of care and some alternative. I'll choose between technically they're called the reference and target, and I'm going to vary one of the properties systematically until you go back and forth. So here's an example. This was done actually by RTI with the FDA. At the time, they were thinking about wearable dialysis devices. They didn't really have the details on it. They just knew that that was the future. So that's why there are a variety of options shown here versus I'm getting hemodialysis in a center. Now, the benefit is I can go home and live my life with this thing. but there's a chance of serious bleeding. Here it's very low, less than 1%. Here it's 8%. Now, if you say, look, you ask people after all the training, which would you prefer? If they say in such a dialysis, then I'm going to make this better. I'm going to decrease the risk here. If they say I'm going to go with the wearable device, then I'm going to increase the risk here to make it less likely. Then I do it 2 or 3 more times, trapping the interval tighter and tighter, and then I stop. You're only looking at 2 properties at a time. It's actually easier than looking at, well, the 6 you saw before. However, I can't see things like interactions. I can't see how these preferences depend on those preferences. Now, half the time, preference studies never really get interactions anyway. They're too noisy. But there are times when it really could be a problem. On the other hand, for certain populations, this is really easy to do, especially say you're dealing with people with limited sight. You can only juggle so much in your head. This is much easier to do in your head. What came out of that study? Well, the results are not numbers, they're intervals because we're tightening the interval, but we never have a threshold. So this is the maximum acceptable risk. Now it's low or high. What was interesting is that it was bimodal. Some people said, I'm not going to take much risk of serious bleeding at all. And other people said, yeah, I'm going to take that, even higher. Why? You were asking about heterogeneity before. Well, we could look at some properties. We could look at whether the person who took the survey was currently using in-center hemodialysis. These are patients. They're doing dialysis. So they know a lot about the disease, or home dialysis or peritoneal dialysis. Now you could see these people had low maximum acceptable risks, 9%. This is high. If you know what peritoneal dialysis, it's not hard to come up with a reason. Here you're sitting, or home you're sitting in a machine. In peritoneal dialysis, you got a plug injecting liquid into your abdomen. You let it equilibrate, take it out, do it again, take it out, do it again, take it out. It's a real bear versus sitting in a much more relaxed manner, reading or hanging out when you're doing those. That's probably what's behind this. At least that's my hypothesis. So anyway, that is part of what's going on in the heterogeneity. Another part will be covariance or confounders, sorry. So big numbers here means I will take more risk of bleeding. Negative numbers means take less. So for whatever reason, and here's your question of why would be important. We don't know why, we just know what. So peritoneal dialysis, a big increase in risk I take. Female versus male, less risk. Low health literacy score, which is a survey that's psychological instrument, quite a bit of a negative drop. Self-management score, your ability to handle yourself with probably handle the machinery, negative score. So again, this is beginning to tell you some of what's behind that bimodal distribution. Now that you know about the bimodal distribution, if you really wanted to understand it, you'd probably design the survey somewhat differently to tighten it up, to understand it better, probably do some of the qualitative research with this result in front of people to dissect it. In fact, that qualitative research might be enough. The threshold technique has been used successfully. So home hemodialysis was one example. So the problem with home hemodialysis machines is you have to have someone there just in case something goes wrong. This company was trying to say, approve it without a care partner. FDA is concerned, well, something could go wrong, like the needle could get dislodged and you could have a fatal problem. So the company with the FDA designed a threshold technique experiment to determine what chance of that happening would be acceptable. And you're looking at essentially a cumulative density function above 30%, at least chance of that happening, over 40% of the people would accept it. Basically, a lot of people would accept a reasonable amount of risk of that event happening, which the FDA said, okay, it's reasonable to put this on the market. The patient and physician will have to collectively decide, but this is telling me that enough patients think the benefits outweigh the risk for a home dialysis machine that's not supervised. Ear tube placement. So here, the idea is instead of having surgery to place the ear tube in a child with infection, there's a gun-like device that would insert it. Very fast, doesn't need surgery, doesn't need anesthesia. Problem is the device didn't always work. And the FDA's concern was, well, if you're going to use this device and it fails, maybe we shouldn't approve it. So what failure rate or what success rate is it required? Now, it's a little strange to me because basically, if the gun failed after 1 or 2 tries, then you could still do surgery, but they wanted it. So the preference study was basically 2 properties, operating room versus the office and the chance of success. It's a real, almost trivial survey. Now, a lot of the survey defined what the operating room was like, this experience, what the doctor's office was like, and ultimately, people said we're willing to accept, about 68% chance of success. Below that, we'd probably go to the surgery. Okay, now you could debate whether that makes sense, but that was done and it was used for approval. This is a threshold technique. You should know that there is a newer variant of the threshold technique called multidimensional threshold. It's being increasingly used because it has the power to look at like 10 attributes, all of which are probabilistic. No way you could do that with contract analysis. You can't memorize, juggle 10, 20 probabilities. But by breaking it apart into little choices like this, you can. Okay, there is an increasing amount of guidance on the preference world. Now, FDA's issued one, the CDRH, the devices group, issued the first guidance in 2016. And it's being updated, but the real thing is in 2026, under PDUFA 7, FDA is going to issue a new, different guidance out of the biologics and drug side. That's what a lot of us in the preference world are waiting for, or fear, depending on how it goes. Meanwhile, ISPOR and the IMI Prefer Public-Private Partnership put out guidances and actually a whole set of recommendations. This is a project that JJ mentioned that I've been involved with, and you can look at the links here. The Prefer thing came out, was really quite well received by both FDA and EMA. EMA has this process called qualification, where they review new methods, new assays, new techniques. They issued a positive qualification opinion on the IMI Prefer framework. That's basically as close as you could get to a guidance, but it's not a guidance. It's another way of saying, if you're going to do a preference study and submit it to the FDA, take a look at this. It'll tell you what they have in mind and what they care about. ICH, the International Conference of Harmonization, is also going to be putting out a new project starting next quarter, and then first quarter next year. They're going to develop guidelines for harmonized standards worldwide for preference studies. A big deal because it's one thing to do a preference study in one country. It's a big problem if you want to do it in multiple countries. Okay. FDA has really acknowledged this importance. They actually have done a number of preference studies themselves, but they're really clear. They don't by themselves, they're not going to determine your benefit risk. It's additional information. And EMA says the same thing. The expectation for this and the balance between where clinical data wins alone or where clinical data is highly informed by preference data should come out of this. They're called additional thoughts. Here actually is a list of some of the examples where the FDA has made a decision based on a preference study. While it's voluntary, these companies did it. And most of these are approvals. And for the most part, these were very successful engagements. Now, what we don't know is how many times companies provided a preference study and it didn't work, but we do know this one because it became a big deal. The Tanuzumab preference study for osteoarthritis was actually excellently done. The company decided to submit it almost at the last minute. It was a mistake because even though the study was well done and eminently publishable, the FDA didn't have a hand in its design. The FDA said, this is a good study, but we would have preferred an opt-out. We would have preferred physician-confirmed diagnoses instead of self-report diagnosis. We would have preferred you define the risk this way versus that way. These are things that can easily have been fixed if the company had talked to the FDA well in advance. But because it was a last-minute decision to submit it, that obviously was not possible. The upshot of this is, if you're seriously thinking about a preference study to support the submission, talk to the FDA at the very beginning, even before the qualitative research, because they'll change your qualitative research in useful ways. And my experience has been, even though only a small number of people in the FDA understand preference work, it's made a big difference and it's worth it. If you're going to do a preference study, it does take time. If you're going to do a rigorous publication quality study, it's going to be at least a year. Takes longer if the FDA is involved, simply because you don't get a meeting when you want. You have these decision points, so on. You need at least 200 people if you're going to do that discrete choice. The threshold, you might be able to get away with 50. A lot of it depends on how many benefits and risks or attributes you have and how hard the problem is. Physicians generally have more consistent answers than patients. You need fewer samples, patients in the sample. And there are lots of ways you can recruit patients. But as I was hinting to before, if you're going to do something for regulatory purposes, increasingly, you want people whose diagnoses are confirmed by physicians. There are a couple of examples where it was glaringly obvious that the patient sample was lying. They didn't have the disease they said. I have one example where people were 20 years younger in one country than another. No way they had atrial fibrillation. So physician confirmed is really the way to go. Okay, preferences can provide a really trustworthy way of getting the stakeholder, patient, physician, caregiver, and so on, perspective on weights, benefits, and risks. And it could supplement or maybe even replace your perspective, the viewpoint of expert physicians who don't have the disease. You can decide whether it's replaced. Not all decisions need it. You saw some of this before. If your benefits are positive, your risks are smaller than the alternative, you just state that and you're done. There are many ways you could do preference studies. The standards, there are definitely a lot of machinery available now, but you could see that there's new stuff coming out in the next few years. In fact, the regulatory decisions and this new preference study guidance that are going to come out will tell you how to do preference studies and before, and you're going to get more position from the FDA on how well, and EMA on how preference studies can be integrated with clinical trial or epidemiologic data to make a decision. Thank you. And that's Carol's talk, so please send Carol a note.
Stand up for 1 second. We're going to just do a stretch. Round. And then give Bennett an applause because I thought he did an amazing job covering Carol's talk. We just found out last night. So thank you so much. And now we'll introduce Bennett to do his talk. So thanks. Were there any, do you want to just do questions at the end of yours? Maybe?
Well, let's see.
Are there any immediate questions?
Now.
So we've got an hour left within the session or less.
Thank you very much. Very nice talk. Have you looked into or are you familiar with the technique as regard to when risk appetite changes, the perception of the risk is changes? For example, the day that Prince died of a fentanyl overdose, that word has been added to the vocabulary of humanity. The reason that I'm asking that we are doing under JJ's supervision 2 studies, it's not about the patient preference, it's about the public preference, and we have an abstract here. So when something happens and people start looking into something, they change the direction of the, or seeing things more important than they used to be. Is there a technique to define this or how to tackle the problem?
It's more a matter of longitudinal studies. So you really brought in 2 issues. One is personal experience with the disease, say a chronic illness. Decision maker has the acute illness, and actually they're the least experienced with the disease. The patient has been ill for several years. They're the most experienced. They're the one whose mind you want to use, but they're not the ones who make the decision. The other issue you brought up is, say, something in the newspaper or public understanding of a disease changes, or a new drug comes out and everybody's thinking about it. Really, you would have to do the study before and after. It's been very rare that people have done longitudinal studies looking at the same people over time. There is one that I don't remember the results for. But as preference work is done more and more budget is applied to it, we will see that. I can't give you any answers to it, but I know that for changes from public issues. But in general, patients learn to accommodate their diseases, and the disutility associated, say, with a stroke reduces over time because they learn to find a way around life, find reasons to live. Their family becomes a better support system. Not always. I've heard of some diseases where it goes the other way. So, in principle, there should be such studies. In practice, I haven't seen enough yet to really answer your question.
Thank you very much. Quick question, maybe a long answer perhaps, with regard to the utility of these sorts of tools for eliciting preferences from patients in the pediatric age range, and that's a heterogeneous kind of range of capabilities. But certainly there's a sort of broad range of age where it's the child and the parents and the physicians' preferences that may differ. So can you comment? I mean, I saw the ear gun
Yeah, that was the parents.
That parents, yeah.
So when you do your qualitative research, you can assess whether the population will work. So you're dealing with children, you're dealing with dementia, you're dealing with psychiatric illnesses, and you study it. For example, psychiatric patients with schizophrenia, when they're stable, have done preference surveys without a problem. And there's a funny story from somebody from your company who was doing pre-testing, and she was terrified to go to the hospital because she was scared of the typical schizophrenia patient. But the physician made sure that the patient was stable when they were testing the survey. Anyway, it was very clear you had to design the survey simpler for schizophrenia patients. It was more of a yes-no property than a probability, for example. And it was hard for them to hypothesize things about themselves. It was easier for them to say, there's another patient with these properties. What would you choose for them? That was easier than imagine you had these properties, these treatments. What would you do? So you can accommodate. For children, young enough, obviously, you have the parents do it. Adolescent, you'll have to test to see whether they're going to be engaged enough to take the time to do the survey. Since most of the time the parent will be making the decision for them, I would think you'd end up with the parent, but it'll depend on the nature of disease. I bet you, for example, ADHD, where a kid will tune their dose daily, they probably should do the survey.
Look, I agree, but there, I guess the reason I asked the question is that the methodologies need to be developed to accommodate for the developmental capacities of younger children. Actually, even children from 7 and above can provide input to quality of life surveys, etc., etc. So I guess I'm making a plea for these sorts of tools and methodologies to, by default, think of that in between age groups. So the very young, the parents, the older adolescents can be more and more involved in their decision making, but it would be great to try and involve the in-between age, because they can have quite valid viewpoints about their preferences.
So let me ask some of the co-speakers if you know examples where people have studied different methods with younger kids.
We can maybe put that back to Carol. And I was also going to say, it also would probably depend on the stakeholder and who's making, where this information is going to be useful. Maybe, right? I mean, is a regulator going to?
I can tell you when we did the IMI prefer stuff, we picked certain methods that were easier to do for certain populations. So for people with a neuromuscular disorder, I forget the details. The discrete choice experiment, where you see those two profiles, too complex. It's just too much information to juggle. So we use some of the simpler techniques where there was less information presented at once. Some techniques are very graphical. Sometimes you actually can use a movie, an animation, to introduce the survey. I'm doing a survey right now in people with limited vision. We have to totally redesign it. We have actually an individual who will be on the phone with them all the time during the survey, and we encourage a caretaker to be with them during the survey. Large font, less animation, and so on.
Yeah, I just had one just curious question. I was wondering if for the discrete choice experiment, like does the order matter in which you provide the choices? And then if yes,
Yes,
how is
they're
that calculated?
randomized.
Oh, they're
Left to
random.
right randomized, question randomized. Occasionally, you'll force a certain question at a certain location for certain testing purposes. But that's actually one reason that electronic patient reported outcome companies find these hard to do. This is not just skip logic, it's very complicated logic.
Thank you.
I'll have to put the mic back up for you. So now I'd like to introduce Dr. Bennett Levitan, who will be talking more about the applications of patient preferences studies to clinical and observational data. This is our last speaker, so we'll continue with the interaction, but we want to be mindful and end on time.
Hi, I'm Bennett Levitan. Okay, so a lot of people want to do a preference study, but they're not quite sure what to do with it or why. So this will talk about the application. All right, so I'm going to start with, what does a company think about when it's going to do one of these things? This is part of a longer talk, but I'll include some of the key points. Why you're doing it, how long it's going to take, and how does that impact your development program? Because remember, we're doing this for benefit risk, not just to get information. Doing it right and how to apply it. All right, so this gets to a point we saw before. If you have an easy problem, you just look at your data and qualitative assessment is enough. Now, as it gets harder, you start wanting to use a benefit risk framework to structure the information, to carefully pick your endpoints with the value tree. And if it gets harder still, you want to do quantitative benefit risk modeling. Now, how often do these things happen? Well, generally, this is what happens. You're either qualitative or in the semi-quantitative realm, which in my view is you're looking at, say, a forest plot or a table of benefits and risks, and your clinical judgment weighs them. But sometimes this happens. And that's where the advanced machinery comes into play. Now, the rub is you don't know where you're going to be until you've got your data, and then you don't have enough time to do this. So preference studies for benefit risk, remember, there are many other applications, but for benefit risk, preference studies are often a hedge. So imagine we have a drug that we know is going to be superior on safety. So it's going to depend, whether you need a preference study is going to depend on efficacy results. Now, if the hazard ratio is less than or equal to 1, which means benefits outweigh, your benefits are good compared to the comparator, we want to land here. And, maybe the preference study would have some use, but you're done. This is qualitatively obvious. Now, if your hazard ratio is greater than, say, 1.1, you may be able to find some roles for a preference study in subgroups, but you're pretty much dead in the water. Your efficacy problem is too bad. Now, if you're in the middle, I'm a little worse in efficacy, but I'm technically non-inferior. But the FDA is really going to say, I care about efficacy. I'm not sure if the improvement in safety overwhelms that. That's where preference results can have a big impact. Now, I'd love to be here as my company. I don't need to worry about anything. But I do the preference study for benefit risk purposes, sometimes as a hedge, in case I end up here. And since you don't know where you're going to end up until top line, you do in advance. Now, there are all those other roles for preference studies. So even if you ended up here or here, you can still take advantage of your preference work, but not necessarily for benefit risk. Now, second thing we care about is timing. If you're going to do it for publication, the things that take the most time are contracting, which we don't think about, but it's easily 3 or 4 months to do a contract in a company like mine. And the design of the instrument takes a long time. Publication can take a while, but you're done. And at that point, the constraints are self-imposed, and it's not too hard to fit this to changing needs. But if you're doing this for regulatory submission, you've got those health authority meetings, and the application to clinical data takes a long time because not only you're doing the math, applying it, but you've got multiple rounds of review by your in-house clinical, statistical, regulatory, and so on staff before they say, okay, we could submit it. Now, that can take so long, especially because these meetings require advanced setup, waiting and then time for them to respond, the health authorities to respond to you, that it can impact your trial timing. And that's one of the reasons you really have to think carefully about how this fits into a trial. If you have a multi-year trial, it's okay. If you got a 6 months trial, it's not going to work, not for a regulatory quality preference study. Now, the other things I mentioned, impact on the development program, that was basically the timing. Getting it right, that's not too difficult because you're typically going to be working with an expert company, and the application is going to be the rest of my talk here. Okay, now there are many ways you can take your raw preference results and turn it into metrics, and you've seen some of these so far. There's more. The complications, the clinical results are heterogeneous. I don't have point estimates only. The preferences are heterogeneous. There's uncertainty in both of them. The endpoints can be dependent. The preferences can be dependent. They're not independent. You know, your covariance matrix, if you did a regression, not everything's on the diagonal. There's going to be a lot of benefits and harms sometimes. Other times there might be one each. And the preferences, we haven't gotten to this, and I'm not going to get into it, but they can be nonlinear. It's worth just giving you a quick example. A 5% change in chance of death from 0 to 5%, huge impact on your decision-making. 5% change from 50 to 55%, almost no effect. Now, once I got that high a chance of death, it doesn't change my decision-making. The weight associated with a 5% change in your attribute depends on where you're starting from. Now, we're not going to show that because it's a lot of machinery and it's complicated, but you should know if you're really going to do this with an expert, they understand that and they'll help you deal with it. If you look at the stuff that came out of IMI Prefer, we thought it helpful to categorize the applications into 3 groups. Looking at the preference information alone, looking at preferences and clinical data together, and then mathematically mixing the two. There are a variety of approaches for each. So we're going to start at preferences alone. So suppose I did a qualitative study. I talked to people, I did a panel or interviews. And this is actually based on a real case study we ran. In acute migraine, you're going to have various measures of benefit. You're going to reduce sensitivity to light and sound. You might remove pain completely, or you might partially remove pain. You might remove it quickly. And then amongst the side effects, I mentioned before, heart attack. Okay, now we had 2 groups. One are people who acted like patients, and another group that acted like physicians. As it turns out, the prevalence of migraine in the US is about 12%. So each group of 20 had a few people with migraine, and they told us what life was like for real. Now, in the patient group, they said pain-free response is unrealistic. There's always going to be some residual pain. You shouldn't include that endpoint. We measure it, but they're saying it's not really something I think about. The physician group said, I can't believe you threw out that pain-free response. We thought if you're going to have the risk of heart attack, you should get rid of your migraine completely. Now, no one's right. It's just a different perspective. Getting to your point, all we've done is identify what's behind their difference in thinking. They're still going to hash it out. Or the physicians will win and the patients will be angry. But that's an example of what you can learn on a qualitative level. Now, you can do the same thing more quantitatively. Fragile X syndrome is a rare genetic disorder for kids. The kids really can't possibly do the preference study. This has to be done by their parents. Now, Genentech was doing this at the time. During phase 2, they wanted to do the study to learn what endpoints they should measure in phase 3. They really didn't know because it was such an unusual disease. And they gave it to family members. Now, these are the 6 properties, attributes: applying new skills, explaining needs, controlling their own behavior. In the sense, it doesn't matter for the story. The size of those purple bars is how important the attributes were. And they all went from not hard to very hard. So they all sort of have the same scale. Now, the bigger ones are most important. The 10 is the most important. The 4.2 is the least important. They went in, and the clinical and commercial people thought the first and the 4th were the most important. The 4th was the least important. The caretakers actually said the 3rd and the 5th were the most important. So the people who went into this had a totally different view of what was important. Now, it happened, the drug failed in phase 2. If they went into phase 3, this is telling us that controlling your own behavior and caring for self would be the properties that you should use to turn into probably a secondary efficacy endpoint. So this is a quantitative way of doing what you saw before. And I think this needed the quantitative. I think qualitatively, you'd have a hard time getting this fidelity. Okay. Maximum acceptable risk. You saw a couple of examples of this before. Here's an old example, but it's impressive. So this is for a treatment that would prevent Alzheimer's. Okay. And you would either you could delay Alzheimer's for 7 years. That was the time frame of the study. Now the side effect is either death or permanent severe disability due to stroke. Now, if you could essentially turn off Alzheimer's for 7 years, people would take over a 30% chance of death or disabling stroke. That's insanely high. What's happening is people are terrified of Alzheimer's, and you're essentially saying, I've got 7 more years of normal life. Now, who would ever approve a drug with a 31% chance of death or disabling stroke? Nobody. But this is telling you that if I did have a 5% chance of death or disabling stroke, patients would be willing to take it. Regulators would never be comfortable, but you get it. But more realistically, you get these are more moderate levels of benefit with more realistic levels of risk. So maximum acceptable risk, it can be a very powerful metric. Okay, next. What if there's several risks? So here, I'm just looking at composite of death or permanent severe disability. There's this new approach that came out in the last few years. You can see the references where we can look at multiple risks and ask what combinations are acceptable. So if I take you from severe to mild symptoms, and this is based on a real example for depression, any combination of adverse event 1 and 2 below that line is acceptable. So instead of just looking at the maximum acceptable risk 8% or 5.5%, I can now more realistically say what combinations are acceptable. And what if that's not the benefit? Suppose I get severe to moderate symptoms. I still have a viable drug. I just have to be below the green line. Even moderate to mild, I have a viable drug. I have to be below the yellow. This is in many ways part of your target product profile. It's basically saying, if I get this drug, moderate to mild, I'm okay. Just make sure the risks are below that. If you get me a drug that has a 3% chance of each of these adverse events, I'll market it, but I have to be below, at least get severe to mild. This is not just a benefit risk thing. This is basically many ways to succeed. It's increasing your path to success. Now, they're not single lines, they're distributions. They can measure heterogeneity or they can measure uncertainty, depending on what you're modeling. I can think in terms of the median on the line, but then I could say the blue line is what 90% of people would accept. So 50% of my population would accept the red line, which may be what I'd use for benefit risk. But more realistically, I'll say I want at least 90% of my population to accept it. So I want to be below the blue line. And that's all doable mathematically. I'm just showing you the concepts. And they could be much more complex. Generally, you get straight lines if the data does not suggest the need for a more complex curve. But if the data suggests it, then you can have fancier curves. And in the reference a few pages back, if you look at these things, you'll see both linear and nonlinear examples. Okay, so that is maximum acceptable risk, but with multiple attributes. In fact, you got 3, the benefit and 2 risks. Yes. Yes, you could do this. When you see some of the calculations, like maximum acceptable risk, choice share, you're essentially doing this type of calculation with all your properties. This is just showing 3, but you can do n-dimensional space. You can't visualize it, but it's the same concept. Oh, no, you could do this with interaction. In fact, this curve can be reflective of interaction between preferences and interaction between the clinical data. It's just, instead of having a simple sum of weights, it's a more complicated function. And that's why we work with those expert vendors, because you don't want to spend all your time disentangling the math. You want people to do this on a regular basis. Now, here's a novel application. Often we come up with a new endpoint and we have no idea what's a clinically meaningful change. So the pro world, patient-reported outcomes have lots of machinery to decide how small a change is meaningful. But preferences give you another way of doing it. Suppose I have this novel time until some event efficacy endpoint. I don't know what change is meaningful, but I do have a heart attack endpoint. And I do know that there's a well-accepted effect size. 0.5% change is meaningful. I'm making it up, but that's the concept. Well, once I've done the preference study, I can say, what is the preference change associated with that effect size? Carry it over here, just the same yellow arrow, and now go back down. This is telling me that a 2-year change in this new endpoint is preferentially equivalent to the accepted effect size here. So basically, if someone tells me I'm happy with this being a meaningful change, then this is a meaningful change as well. We use this to show how fatigue in multiple sclerosis compared to relapse rate, which otherwise, conceptually we want one bigger and one smaller. This told us exactly how one compared to the other. It's actually the same math as maximum acceptable risk, but used a different way. So those were a couple of examples of the ways to use preferences alone. Now, how about preferences in clinical data used jointly? These are primarily graphical techniques. So here's one that was actually used at an advisory committee meeting for atrial fibrillation. It's a forest plot. Anything on the left favored the study drug, anything on the right favored the comparator, and the endpoints are in order of weight, where the most impactful are on top, least impactful on bottom. Now, it's kind of obvious just glancing at it that the endpoints on the top and middle either favored the study drug or are on the line. It's only the ones on the bottom that favor the comparator. This was enough. You didn't have to do any math to combine it. Basically, the study drug helped where it was important and hurt where it was less important. And I could tell you the current head of the FDA presented this slide, which was kind of funny because he actually had trained about a third of the people on the advisory committee panel. But this went over very well. And it was even written up in the trade press as a way of bringing the patient perspective into benefit risk. Now, you can get fancier. This is just the rank of the weights. You can sort them by, position them by the size of the weights. The benefit is you could really see there's a big gap between the ones on top and bottom, which helps strengthen your argument. The problems are twofold. One, rank is easier to defend than weights. Here, you could probably go by intuition half the time. Here, you will have to defend it with a preference study or some utility methods. And the second, just graphically, they can get on top of each other. Sometimes you could just do this. You could do a table, an effects table. It's drug 1, drug 2, the risk difference or the rate difference, the graphic of the rate difference and the weights. And I'm beginning to weigh them in my head. Okay, this is the biggest weight, this is the second biggest weight, and so on. And it's the same argument as before. The big weights are generally in favor of the things that are on the left, and the low weights, the ones on the right. But you can kind of get a sense of where I'm going. It's not always going to be so easy. Suppose this one was on the right, the one on top. That's where you need the mathematical machinery to combine those. Now, before I get into that, and it'll be the same multi-criteria approach that JJ said, I want to take you on a bit of a detour. Think about the endpoints you're applying this to, or rates. There are statistics and epidemiology. They can give you in proportions, they can give you exposure time rates, they can give you Kaplan-Meier rates. Well, what's the right thing that a preference weight applies to? The weight doesn't change, but your numbers change. So let's think about progression-free survival and say for an oncology treatment. So the curves might look like this. Well, what do I want to use? Well, incident proportions, they're easy for people to understand, and they most generally align with the concept of a weight per event, but they ignore the timing of events, exposure time, discontinuation. Exposure time rates, they're harder for people to understand, and they're not as well aligned with the concept of a weight per event, but they account for all this. Kaplan-Meier rates, well, they're great for the timing of events, and they're easier to understand than exposure time, but you got to pick a time. And there's a bit of a arbitrary aspect of that. Plus, there are multiple time points, and we actually often do all of them. Well, let's go back to that PFS curve. If I pick time 1, I get that difference between them. Time 2, time 3, time 4, all differences. I can get different results depending on the Kaplan-Meier rate time I did. And incident proportions, exposure times also would be different. Well, my argument here is these two curves are not showing a difference to rate. These are showing a difference in the time to progression. You are better off actually having a preference study which looks at the delay of progression, not the rate of progression. Now, if your study ended at time T2 and that was it, yeah, that's fine, and I've done that. But if your study looks like this, T4, everything is collapsed to the same. Your benefit really is delaying the progression. This is my point of saying that you got to think really carefully what your clinical data is to which you can apply a preference study before you apply your preference study. Because naively, you might think proportions, but your clinical data, the stuff you're going to bring to the health authorities says something different. Okay. Preferences and clinical data mathematically combined. And you could see some other methods. So choice share you saw before, so I won't get into it much. Choice share is basically saying, if I give you treatment A and treatment B, what proportion of people would pick one over the other? This is actually from the study the FDA did on obesity devices. And these are the calculations that were used to help in the approval of Enteromedic's obesity device, sort of a pacemaker for the gut. And by changing the nature of the device or having a gastric band, you could see different proportions of people would pick it versus none. Since it was an elective device, having any reasonable proportion of people who would choose to use it was enough for approval, to support approval. But I'm going to get more into the MCDA, which is more common for us. Okay, the value tree is going to be really important here, the set of endpoints you use. Trial endpoints can be fine individually, and they often are, but they don't necessarily work together collectively for benefit risk. They can double count. They can be, some of them can have very broad clinical impact. The concept of weight applied to something that goes from death to a minor disability doesn't make sense. They can have this preferential dependence, which I'll explain. They could be dependent causally. A could cause B, could cause C. Why do I need to look at all three? And then the type of measures, I just went through that. So you need to revise the value tree to avoid or some of these problems are accommodated. Now, there's some work on this. There's that task force from ISPOR that you heard about has some guidance, and these are the references for it. You could see they have steps to think about when doing essentially MCDA. And that was mostly what the quantitative benefit risk assessment dealt with. So I won't get into it, but the details of how you might do this are worked out in there. But I'm going to show you an anticoagulant example for acute coronary syndrome. So in ACS, you either have unstable angina or a heart attack. You're going to get thrombin for a while, at least for 6 months, and it's going to increase your chance of getting ischemic stroke, an MI, heart attack, or death. And at least at the time, you would use aspirin or an antiplatelet agent. But even with those, you would still have a good chance of an MI or death and so on afterwards. So let's do a mock study where people with a recent acute coronary syndrome were randomized 1-to-1 to a study drug or placebo plus standard of care for 2 years. And the goal is to show superiority in the composite of cardiovascular death, MI, or stroke. It's very common. So our primary is that composite. The primary safety endpoint, which you do test for in a typical cardiovascular study, is TIMI, major bleeding. There's several different standards for major bleeding. This one is the composite of a fatal bleed, symptomatic intracranial hemorrhage, or bleed with a hemoglobin drop less than 5 grams, greater than 5 grams per deciliter, and we measure everything else. But I'm just going to think of these two. Now, here's the primary efficacy and primary safety broken up and color-coded by whether they're ischemic, hemorrhagic, or mixed. Take it one step further. The stroke could be ischemic or hemorrhagic. The death can be ischemic or hemorrhagic. Under the primary safety, I can have a fatal ICH or a non-fatal ICH, which overlaps tremendously with a hemorrhagic stroke and hemorrhagic death. In other words, the very same event can count both as an efficacy event and a safety event. In fact, I could turn the benefit risk problem to favoring one drug or the other just by varying the degree of overlap. Now, you need to disentangle this to do benefit risk quantitatively. So, okay. Now, another problem is the broad clinical impact. Every endpoint has some range of impact. even death. It could be a slow lingering death or a sudden death. Composite endpoints can be especially concerning. So we'll look at TIMI major bleeding. So a fatal bleed and symptomatic ICH, those are on the upper end. But a bleed associated with even as large a drop as that, well, the ICH could be fatal and irreversible, but hemoglobin drops, they could be reversible with no sequela. In fact, in one study, it was 75% of them were not fatal and were not irreversible. Only 25% were fatal or irreversible. Putting them in the same bucket makes it very hard to do benefit risk. You really should disentangle them. So this is what the value tree looks like in the SAP. So cardiovascular death, disabling stroke, disabling or non-disabling stroke together, MI, TIMI major, TIMI life-threatening, TIMI minor bleeding, different levels. Okay, so the ischemic and hemorrhagic events are mixed because they could both be here, they could both be there. I'm double counting a fatal bleed in ICH under both benefit and risk. I showed that before. But not only that, even within efficacy, I'm double counting of death. Cardiovascular death can include a stroke death and an MI death. So not only is there double counting between benefits and risks, there's double counting between benefits. And then there's that wide range of major bleeding. You can redesign the value tree to remove those problems. So here I've got CV death that's not hemorrhagic, fatal bleeding down here. So there's no bleeding double counting. These are non-fatal stroke, non-fatal MI, and only ischemic stroke. So I have death here, not there, and not there, and I only have non-bleeding here. I only have bleeding down here. There are other ways to do it, but this way solves those problems. Ischemic and hemorrhagic events fully separated, no double counting of events between benefits and risks, no double counting within the benefits, and I've separated the clinical impact here. Fatal bleeding, and ICH that's not fatal, and everything else. Everything else is typically a GI bleed. There are other ways to do it, but now I'm much more set to go into benefit risk. The other things here, well, I mentioned them before. Preferential dependence means when I think about this endpoint depends on the value of that endpoint. For example, do I care about a rash if I'm talking about cancers? Not too much. Do I care about rash if I'm talking about migraine? Yeah, because I've got a long, healthy life for the most part. My weight for rash is going to depend on the chance of death. Now, technically, that gets in the way of the models we're doing. You can avoid it in certain ways, but basically it violates some of the assumptions we're doing. It's probably more technical than needed for this audience, but I wanted you to be aware that there are more things to know that your preference experts will help. Causal dependency. Suppose a rash causes excoriation. That could lead to double counting because the weight for excoriation probably assumes that you had the rash first. I don't want to include both rash and excoriation. I probably just want to include excoriation. Or I might say excoriation without the rash. Finally, statistical measures. So here's a fun one. Do I look at events on a person level or an event level? I can look at the proportion of people with a rash, or I can count the rate of people with rashes, allowing for more than one rash per individual. If I have multiple rashes per individual, is the weight for that, say person has two rashes, is the weight for that the same as two people with one rash each? No, but you're not going to get that difference unless you set things up for it. And generally, the preference literature doesn't have that fidelity. So those are some of the complications. Okay, now the example. This is a forest plot for a mock cardiovascular study in acute coronary syndrome. Anything to the left favors the study drug, death, CV death, non-fatal MI, non-fatal ischemic stroke, favoring by far out of 10,000 patient years, 10 fewer deaths, 29 fewer non-fatal MIs, 104 fewer non-fatal ischemic strokes, no difference in fatal bleeding, meaningless small drop in non-fatal ICH, small increase in the GI bleeds. You don't need a quantitative model. You look at this, if you understand the endpoints, benefits outweigh risk. There's no way that's anywhere near as impactful as ischemic stroke and MI. However, suppose I move death to the other side. This is harder because now I'm going to trade off a non-significant increase in cardiovascular death versus a significant drop in non-fatal ischemic stroke and a little bit non-insignificant MI, that's hard. That's where quantitative model can help. And that's actually one of the places the FDA most uses MCDA in cardiovascular. Now, the math, I wanted to give you a flavor of it. Basically, you saw this before with JJ. I'm going to take the rates and multiply them by a weight. It can get more complicated, but roughly the score is equal to the sum over every endpoint of a weight for the endpoint times the rate. I'm really going to just care about the difference between them. So ultimately, it comes down to the risk difference, the difference in the rate or the risk of the event between the two arms times the weight summed up over all the endpoints. The way they understand this is that every weight is positive, and in cardiovascular, every event is a bad thing. You don't want it to happen. So the more events, the higher the score, and basically lower scores are better. So the bigger the score, the more bad things happen to you. That's all to know. Okay, so here's how it could work. If I have a simple example with cardiovascular death and non-fatal MI, I'm going to just look at the risk differences. For 10,000 patient years, there are 10 more deaths. Now I'm going to normalize the weights. So death is a weight of 1. So the contribution is 1 times the 10. 10. The weight for a non-fatal MI is 0.36 relative to death. So I have 100 fewer non-fatal MIs, multiply that, minus 36. Now I add that together, I get 26. What does that mean? Well, remember, the weight of death is a 1. Every other death is normalized to that 1. So I can interpret this as the equivalent of 26 fewer deaths per 10,000 patient years. That's the advantage of this normalization to 1. Even though the math can get a little frightening or hairy, at the end of the day, this tells me that the difference between these two treatments is the equivalent of 26 fewer deaths per 10,000 patient years, which makes it much easier to interpret than some of the other methods. So now a real problem is going to have lots of endpoints. I'm going to have differences and weights. I multiply them, I get a score. Just like this is the equivalent of 26 fewer deaths, this is the equivalent of 15.9 fewer deaths per 10,000 patient years. There's a confidence interval. There's a probability. I'll explain where this comes from in a second. But just from a point estimate perspective, the study treatment is better than the placebo by the equivalent of, let's call it 16 fewer deaths per 10,000 patient years on placebo. So this is what it looks like if I take those contributions and graph them. So it's pretty obvious that the main drivers are CV death versus non-fatal ischemic stroke. These are all the components, and that's where they add up. Now, do I trust this? Well, that's where the sensitivity analyses come in. First thing I could do is ask, if I vary each weight, does it change the answer? Because the weights are not right, they're approximate. I'm going to take each weight and vary it by plus or minus 2 standard deviations. And behind this is the assumption that I have distributions for weights. They're not just single numbers people came up with. You get them from preference studies. So if any, this is the answer, minus 16. If any change in the weight causes me to go past 0, I flipped to favoring the other treatment. And you can see there's only one weight that can cause that to happen, the weight for non-fatal ischemic stroke. It can take me way over to the right. That's not a surprise. If you look at the differences here, the non-fatal ischemic stroke has the biggest risk difference. So a weight there can have a big impact. Okay, so can I trust this? So I can do mathematical modeling, a Monte Carlo. This is that difference between the multi-criteria model and that stochastic multi-attribute acceptability analysis. You need to say that to graduate this course. Basically, I take every input, both the clinical data and the weights, and I vary them randomly. I use the same distributions for the clinical endpoints that came from the clinical trial. Stats does that. And I use the distribution for weights that came from a preference study. And I look at that MCDA score, or in this case, the difference. Anything to the left favors the study drug, anything to the right favors placebo. You could see that more of the probability mass is to the left of 0. If I do a cumulative density function, the point where it's 0, 65%, 65% chance benefits outweigh risk. This is not a slam dunk, but this tells you that I'm helping more than hurting. And I picked this example on purpose because That's sort of the middle bet, hedging your bet. This is basically saying using all the information I've got as best I can, taking into account statistical uncertainty on my weights and clinical data. This is my assumption of whether benefits, demonstration of whether benefits exceed risk. Now, this is just for 2 drugs. You got a little hint of this from JJ. What if I have multiple drugs? So this is from that ISPOR example. Here are 4 different MCDA scores, and the bigger the better. And you could see that this is for psoriasis, brodalumab at 210 milligram has the highest score, mostly because of the PASI 75, your skin clearing. Placebo does terribly. The others are, good, not quite as good. Now, do you trust it? Well, there are all sorts of analyses you can do. One is that Monte Carlo. Now, this Monte Carlo only looked at the, didn't look at both the weights and the rates. It only looked at one of them. But the type of plot is what I wanted to show you too. This is the probability that Bro 210 was the first ranked result. Like the same as the probability that I'm on the left versus the right. 97% chance that this is the best. 3% chance that it's second. Now look at the others. Now let's just say almost half and half, second and third, and placebo, 100% chance it's the fourth. When you have multiple alternatives, there are ways to take that same machinery and interpret it. This is actually pretty good. If you were looking at this, this would give me a lot of confidence that I can trust that Bro 210 is the best choice. Now, it's a gimmicked example, but in real life, if you see something like this, that's great. Take-home messages. So MCDA and its probabilistic equivalent SMAA, it's a powerful technique. It lets you deal with numerous benefits and risks, have a lot of uncertainty. And it could show you the degree to which each endpoint impacts the benefit risk balance by this, the degree to which each component contributes. You got to look at that value tree before you do this. If you have double counting, this math doesn't make any sense. And if you do a preference study with double counting, you've wasted hundreds of thousands of dollars. The simulation is often necessary to basically trust the results. You can use a simpler approach, and I showed you a simpler approach. Remember that value function JJ was talking about? I didn't use a value function. And if you go into the mathematics behind this, I can explain why. But you can get away with that. In this case, it was because every endpoint was a probability. I wasn't mixing probabilities with times, with high, middle, low scores. That's what the utility function will do, or value function. Converts them to a normalized scale. Here, everything was a rate, and everything was a bad thing. So I just used simple weights times the rates. Are we all set? Can we just do something like calculate the maximum acceptable risk? Or can we do a MCDA score? Well, it's not always straightforward. You saw some of this. If the clinical results are heterogeneous, you're probably going to want to do the model in different subgroups. If your preferences are heterogeneous, you're going to want to associate preferences for each subgroup with the data for that subgroup, which means you have to go out of your way to get a large enough sample to represent them to each subgroup. The clinical results uncertainty and preference result uncertainty I showed you, that's how you get these distributions. Dependency between endpoints and between preferences, that you can do, but it's behind the scenes. The Monte Carlo can't draw independently. The Monte Carlo has to use those complicated covariance matrices. All doable, is just messier. And you can't just look at your weights sometimes. Sometimes you're going to have to deal with the weights being nonlinear. In the examples we showed you, the weights were the same no matter what. But if your endpoint's only near the 100, if I'm only around here, I can get by with a linear weight. But if I'm dealing with a wide range, remember that thing that a 5% difference here is important, here it's not? I have a wide range of my attributes in the different choices, it will be important. So you can't just say I'm going to use the easier approach without having some sense of your clinical data. And many benefits and many harms, you can do that. You just have to have the machinery to add all those endpoints together. What are my thoughts? So I showed you preferences alone, preferences and clinical data viewed jointly. the visualization techniques, and the mathematical combination. So this is my view. It's not official. These have the lowest flexibility, but they're very easy to do, easy to communicate, very transparent what's behind it. I think the health authorities are comfortable with it because it's so transparent, but it's not going to solve the problem all the time. Showing them together has more power because you're connecting that data to one set of data to the other. It's not hard to do, it's a little harder to communicate because you're now mixing more data together. It's very clear. I think because you're looking at the clinical data but not mathematically merging them, it aligns with the way the FDA or the EMA think about it. Now, if you mix it mathematically, it's very powerful. It's very more complex. It isn't that complex for a mathy guy, but for clinical people who are not used to it, it's new. Sometimes, it's actually not as hard as most statistics, but statistics gets a pass. People are used to it. Benefit risk machinery, decision analytic machinery is new. It's harder to communicate. It's less transparent. The health authorities it's beginning to gain traction. As you heard earlier, the FDA put out their first MCDA in cardiovascular about a year ago. They've asked companies sometimes to do MCDA. EMA likes the concept of MCDA, but doesn't think it's needed all the time. So I think it's going to get more and more traction. Takeaway messages. There are lots of ways you can use patient preference information to inform decision. Some of them are going to require that you rethink the endpoints for issue to avoid making benefit risk misleading or less meaningful. I try to use the simplest approach, like I'll use preferences alone if I can get away with it, or this. I will try to avoid that, not because it's hard for me, but because it's going to be harder to defend it to other people. Often I actually use multiple approaches. Like I'll never do a quantitative model without first looking at the data by itself and the preferences by itself. So in a sense, if you do the ones on bottom, you have to do the ones on top no matter what. The mathematical combination approaches, the MCDA, generally they were in the academic literature, but you're beginning to see the health authorities start to use them. As I said, we like to use the combination approaches. And if you're going to do this in the real world, you got to take the heterogeneity, the preferences of clinical data, and nonlinearity. At least you got to think about it. And that's where the experts in preference you work with are most important. Okay, I see I'm at 42 minutes. Wow. Happy to take questions, or if you want to talk to Bennett Levitan from the prior talk, he'll also be happy to answer questions.
When you had to avoid double counting, you had to restructure the whole value tree. I mean, in this situation, how can you make it relate then to the original value tree where all the probability and primary endpoints and all the write-up was based on? Like, how do you make that connection between the two?
I usually have this, we'll write a benefit risk SAP, statistical analysis plan. We'll start with the SAP from the primary trial, pivotal trial, but then we'll modify it to meet benefit risk needs. Then we work with the statisticians. They almost always correct it. We make sure that we're not creating endpoints that can't be calculated. And they will calculate anything we need, but we're also not trying to do hypothesis testing. Efficacy, sometimes safety, will have pre-specified hypothesis tests that have to be proved to get your drug approved. By the time you're at benefit risk, you don't need to do that anymore. And it's very rare that you're going to have a testable hypothesis in benefit risk, usually because you have to have your weights trusted in advance. So at that point, it doesn't matter to me that I'm using endpoints that were not in the primary and secondary, as long as they can be assessed from the same original assessments.
In telling the story, you will not find any issue or difficulty in telling the story about your drug and the benefit risk using outcomes that were not really in the primary kind of.
We've had some very formal statisticians push back. So we did something in in depression. Now, in depression, you use a clinical reported outcome measure called MADRS. And it's a 10-point instrument, each with like 5 or 7 levels. So maybe there's 70 different scores. It's very hard to know what, say, a 4-point change on that means. But what people do understand is a halving of the score is considered a response, and having a score below 10 is called remission. Basically, you have normal mood. Now, we used remission and response in our benefit risk work. The statistician said, that's not your primary efficacy endpoint. Why are you using it? Everyone in the AdCom understood it. It was meaningful to them. It was meaningful to patients. So we basically went off the primary SAP for benefit risk because we got interpretable endpoints.
That's very good. One last addition. Then what about the resistance you might get, because people can say, well, that's all fine, but you cannot get a claim on these modified endpoints. Is this also a challenging point?
At this point, benefit risk rarely has a real role as a claim in the label. You have to work real hard to find a claim that has any benefit risk information in it. They'll say benefit risk, but it's a conceptual notion. In fact, labels just have a benefit section and a risk section, or efficacy and safety. But when, to do benefit risk, you have to have both efficacy and safety together. We got pushback. You're putting your safety data in your efficacy section. Or another time, we put in risk differences, like on the forest plot, and someone said, the regulator said, we don't take that NNT stuff here. There has to be a reconsideration of how benefit risk has a role in labels. The FDA is aware of that because companies like mine have raised it, but it hasn't happened yet. And then when that happens, the issue of stuff that's not in the primary SAP will come up.
I'm cognizant of the time and very appreciative of all of you sitting here, but are there any other questions that anyone in the audience has? I just have one question, because we were going to do a panel discussion, but we're closing up. But since this is a International Society of Pharmacoepidemiology, I would like to ask the panelists for a 30-second answer. That's it. How have you, if you're an epidemiologist, participated in these benefit risk or preference studies? Or how do you see pharmacoepi participating, or how have you interacted with pharmacoepi folks?
Well, I'm actually in an epidemiology department. Benefit risk in a post-approval setting is very different. First, you obviously have much more data, many more patients, and you can look at data longitudinally, but you don't have anywhere near the resolution. So I'll have fine-grained details in those value trees for trial data that I cannot reproduce except in specialized databases. In death, for example, if I don't have death, I'm really stuck with cardiovascular benefit risk. It's been done, but nowhere near the fidelity. That's one of the biggest differences. I'll leave it at that.
Anyway, I think you have multiple questions, but I'll answer quickly. As far as my involvement in benefit risk, in my tenure at Sanofi, actually, I built maybe 6 different teams, which mean also bringing the technology. So first I build the epidemiology team, epidemiology analytics, which means we do all epi studies in-house in terms of using existing data sources. That's what we're one of the first in industry. And then we start benefit risk evaluation discussion by publishing that paper and got involved in many areas. And I built signal detection team, 2012, and then benefit risk evaluation team as a team, 2017, and what we call it global safety sciences, which is combination between epidemiology, benefit risk management, signal detection, and so on and so forth. That's the connection. I think it doesn't matter what your field is, it could be connected to benefit risk. Now, in my mind, as an epidemiologist, that's my background, I actually make it easier to see the importance of benefit risk as a structured and quantitative approach, and epidemiology feeds into it. It's not identical, but I think the role of epidemiology could be big in every aspect of benefit risk. I guess there's 2 minutes, but sorry. I know.
Yeah. Do you have anything to add, Tarek?
I'm not going to add much to what my colleagues said. I'm not going to repeat it, but I wanted to be aware that one of the challenges of the role of pharmacoepidemiology in benefit risk is where the benefit risk activity sits in the company. Like in my company, it sits under safety. I'm in safety, and in Gigi's company, it sits under epidemiology. Same for Bennett. So this will sort of shape the contribution and the way you contribute into the benefit risk. So you have to be cognizant of that. You might have to do a little bit more of an effort if you are in a company where it sits under different divisions. So that's all.
And I'll just add, worked with several different companies, and these companies are very advanced because they have experts in sitting there that know how to do it and have been involved for many years. In companies where they don't have a lot of people that have the experience, the epidemiologists that they're familiar with it can have a role and definitely needs to have a seat at the table. It's definitely a multidisciplinary approach. And I think the biggest challenge is trying to figure out who the decision makers are and how to get people on the same page. So we didn't get into the details of exactly of the BRAC framework, but the decision context and just talking through that and getting everyone on the same page early on, and even just filling out, I think just the first couple of steps and seeing where the gaps are very important, and then just taking it along. So I'll just end here and say thanks to the faculty for all your time to put all the slides together, and to Bennett for studying an extra set of slides last night especially. And to all of you again, thanks so much for sitting with us today, and I hope you have a great rest of the conference.